Kiran, good morning.
Very good morning. I am the second participant.
Last time కూడా అంతే.
అవును.
ఆ మధ్యలో ఆ బజ్ సౌండ్ ఏదో వస్తుంది కిరణ్, ఎందుకు వస్తుందో?
అవి అంటే ఇప్పుడు నేను ఆన్ లో పెట్టున్నాను కదా సార్.
కిరణ్, గుడ్ మార్నింగ్.
గుడ్ మార్నింగ్ సార్. నేను ఆన్ లో పెట్టినాను కదా. సెకండ్ పార్టిసిపెంట్.
ఒక్క నిమిషం, ఏం లేదు, యూట్యూబ్ లైవ్ కూడా ఇచ్చాను.
రీ రికార్డ్ అవుతుంది.
అది కొంచెం పాజ్ తీసుకుని రిపీట్ చేస్తుంది, అందువల్ల వస్తుంది.
అదే
నేను ఇక్కడ యూట్యూబ్ క్లోజ్ చేస్తే సరే.
అవును సార్. సౌండ్ అన్నా తగ్గించాల.
కొంచెం బయట రోడ్డు మీద క్యాచ్ చేస్తున్నట్టుంది.
లాగుతుంది.
ఇంకా సంగతిలో
వేర్ ఈజ్ ఆర్గనైజర్
10:45కి చేరతాడు.
అవును, అప్పటిదాకా ఏం చేస్తాడు?
నేను స్టార్ట్ చేసేస్తా 10:30 షార్ప్.
అవునా?
అంటే అది అలవాటు, అంతే, ఏం చేయలేము.
అదే సరేలే.
ప్రసాద్, గుడ్ మార్నింగ్.
మన వాళ్ళందరికి ఇన్ఫర్మేషన్ వెళ్ళిందా? సైన్స్
గ్రూప్ లో మెసేజ్ పెట్టాను.
ప్రోగ్రాము
ఇది అంటే నేషనల్ సెమినార్ అని ఒక 10 రోజుల కింద. ఇదంతా యూట్యూబ్కి వెళ్ళిపోతుందా మనం మాట్లాడేది?
వెళ్తాం సార్. అక్కడ దాని లైవ్ ఆప్తేనే వినిపిస్తా ఉంటారు.
అదే
లేదంటే ఆపేయండిలే టాక్స్.
ఒక నిమిషం, లైవ్ పాజ్ తీసుకునేది ఉందేమో చూస్తా.
చూసి
రికార్డింగ్ అయితే పాజ్ చేశాను.
రికార్డ్ పాజ్ చేయొచ్చు, బట్ లైవ్ లో వెళ్ళిపోతా ఉంటది.
లైవ్ ఆపలేము,
ఆపలేదు.
తర్వాత మాట్లాడుకుందాం.
Good morning, madam.
Good morning, sir.
Chakrapani garu.
మేడం నమస్తే. మేడం
Namaste, sir.
ప్రసాద్ సార్ పిలిచారేంటి?
సార్ Chakrapani సార్.
సార్ చెప్పండి.
కోఆర్డినేటర్ గారు ఏది?
వస్తారు.
మీరు ఆర్గనైజింగ్ సెక్రటరీ కదా?
ఆర్గనైజింగ్ సెక్రటరీ షణ్ముగం సార్.
మీరు కోఆర్డినేటర్ కన్వీనర్. యూట్యూబ్ కూడా ఇచ్చినట్టున్నారు.
ఇచ్చాను.
ముందు అక్కడ వెళ్ళి ఇక్కడ వచ్చాను.
అవునా?
ముందు యూట్యూబ్కి వెళ్ళి మళ్ళీ ఇక్కడ వచ్చాను అంటున్నాను.
అదే అంటే ఇఫ్ ఒకవేళ 100 ఎక్సీడ్ అయిందంటే జాయిన్ అవ్వలేరు. 70 దాకా వచ్చినట్టుంది రిజిస్ట్రేషన్.
అదే యూట్యూబ్ వన్ వే కదా?
అవును.
అయితే టూ వే.
శ్రీనివాస్ సార్, గుడ్ మార్నింగ్ సార్.
సార్ మార్నింగ్.
గుడ్ మార్నింగ్ మేడం.
షణ్ముగం సారు ఏదో ప్రేయర్ సాంగ్ పాడమన్నారు.
మ్మ్.
కంపల్సరీ పాడాలంటారా?
కంపల్సరీ మేడం.
అరే, కావేరి మధ్య దేశే బదులు పినాకిని మధ్య దేశే అంట, పర్లేదుగా? రంగనాయక స్వామిది సప్త ప్రకారం వచ్చే అది గోడ మీద రాసి ఉంటుంది కదా, నెల్లూరు రంగనాయక స్వామి గుడి మీద, పినాకిని మధ్య దేశే.
మీకు ఏది కంఫర్టబుల్ గా ఉంటే అది పాడండి.
అంటే మధ్యలో మీరు ఏం అనకూడదు మరి, అంటే దాని మీద కామెంట్ చేయకూడదు ఇంకా.
కానీ
సరే సార్ అయితే.
నాకు
ఎట్లా పోతుంది?
శ్రీనివాస్ సార్, గుడ్ మార్నింగ్ సార్.
వాసుదేవ మాస్టర్ గారి గురించి
Professor Rajsekhar sir, welcome sir.
Good morning, Shekhar Pandey. Am I audible?
వినిపిస్తోంది రాజశేఖర్.
Okay.
సో కోహోస్ట్ ఇచ్చేస్తాను, షేర్ చేసి అదే పవర్ పాయింట్ అది షేర్ చేయడానికి అవుతుంది కదా.
Okay. కెమెరా పనిచేస్తుందా?
లేదు,
సమ్ ప్రాబ్లం ఉన్నట్టుంది ఇందులో.
క్లియర్లీ ఆడిబుల్,
ఓకే ఫైన్.
విజిబుల్ గా లేదు.
అందరూ జాయిన్ అయ్యారా?
అవుతూ ఉన్నారు.
ఓకే.
డాక్టర్ శ్రీనివాస్ కూడా ఉన్నారు. మా ప్రిన్సిపల్ మేడం డాక్టర్ బి సుజాత ఇస్ ఆన్లైన్.
ఓకే.
And other faculty from our institutions.
ఓకే.
And then Dr. K.R. Shah వారు కూడా ఉన్నారు.
ఓకే.
సార్ గుడ్ మార్నింగ్ సార్.
సార్ గుడ్ మార్నింగ్ సార్.
అదే ఇందాక ఫోన్ లో మైక్రోఫోన్ పర్మిషన్ లేదు, ఇచ్చేసరికి లేట్ అయింది.
ఓకే సార్.
ఓకే సార్, నేను ఉన్నాను.
మీరు కూడా కోహోస్ట్ ఇచ్చేసి ఉన్నాను సార్ ఆల్రెడీ.
చేస్తా అక్కడ వెళ్ళగానే బస్
అలాగే
స్టాప్ లో దిగాను, కాలేజీకి వెళ్ళగానే వీడియో కూడా ఆన్ చేస్తాను.
ఓకే సార్. వెయిటింగ్ ఫర్ డాక్టర్ షణ్ముగం.
గుడ్ మార్నింగ్ సార్.
మార్నింగ్ ఫోన్ చేశారు
సార్. షణ్ముగం కూడా ఉన్నాడు సార్.
ఓకే సార్. మాస్టర్ గారు గుడ్ మార్నింగ్, షణ్ముఖం గారు గుడ్ మార్నింగ్, గుడ్ మార్నింగ్ అండి, సుజాత మేడం గారు కూడా
గుడ్ మార్నింగ్ సార్. గుడ్ మార్నింగ్.
కేఆర్ షా అంటే ఎవరు అనుకున్నాను నేను.
ఒక 2 minutes సార్, ఎందుకు జాయిన్ అవుతున్నాడు సార్, ఇప్పుడు మెసేజ్ పెట్టాను.
So meanwhile, shall we start?
జాయిన్ అవ్వండి రూమ్ లో. ఇద్దరు చూసారు.
So meanwhile, I will be announcing the program. Welcome to this national seminar on Frontiers in Biological Sciences 2026, conceptualized by the Department of Zoology of our institution, the PRR and VS Government College, Vidavallur, from SPSR Nellore district. So under the able leadership of Dr. B. Sujata, our principal ma'am, we designed this program, the lead role being taken by Dr. Shanmugam. And the program is as follows: initially prayer, followed by address by our principal ma'am, followed by a brief uh, remarks by conveners, and then the organizing secretary, followed by presentations from Professor M. Rajasekhar, Department of Zoology, SV University, and then Professor LVKS Bhaskar, Department of Zoology, Guru Ghaziadas Viswavidyalaya, Chhattisgarh, and then Professor V. Sivakumar, Associate Professor, ISER Tirupati, and then lastly by Dr. S.B. Sainath, Department of Biotechnology, Vikram Simhapuri University. We have sandwiched oral presentations at 12:30 to 1:30 p.m. of different participants, other delegates. And the program will end by concluding remarks from our principal ma'am and vote of thanks by the organizing secretary, Dr. K.R. Shanmugam. And over to Dr. Varalakshmi for prayer.
Thank you, sir. सप्तप्रकारमध्ये सरसिजमुकुलोद्भासमाने विमाने पिनाकिनी मध्यदेशे पणिपतशयने शेषपर्यङ्कभागे निद्रामुद्राभिरामं कटिनिकटशिर पार्श्वविन्यस्तहस्तम् पद्माधात्रीकराभ्या परिचितचरणं रङ्गराजं भजेऽहम् रङ्गराजं भजेऽहम् राजं भजेऽहम्. Thank you, sir.
Okay, thank you, madam. And then initial remarks by our principal ma'am, Dr. B. Sujata. Over to you, ma'am.
Respected faculty members, esteemed resource persons,
It seems that there is a glitch here, and ma'am will connect soon. మేడం మీరు మ్యూట్ అయిపోయారు, అన్మ్యూట్ చేసి మాట్లాడతారా? Ma'am, you are not audible, ma'am.
Hello.
Now it's okay, ma'am.
Respected faculty members, esteemed resource persons, dear colleagues and students, a very warm and pleasant morning to one and all. It gives me immense pleasure and a deep sense of pride to welcome you all to this national seminar on the theme Frontiers in Biological Sciences. The significant academic event is being organized by the Department of Zoology of PRRVS Government College, Vedavalur, in collaboration with the Department of Zoology, Government College, Ramachandrapuram. At the outset, I extend my heartfelt gratitude to all the dignitaries, scholars, and participants who have joined us today from the various parts of the country. Your presence adds great value to the seminar and inspires our academic community. Biological sciences, as we all know, are evolving at an unprecedented pace, from molecular biology to environmental conservation, from genetics to biodiversity. The frontiers of this discipline continue to expand, offering innovative solutions to global challenges. This seminar provides a wonderful platform for intellectual exchange, collaboration, and exploration of new ideas that can shape the future of science and society. I sincerely appreciate the efforts of the Department of Zoology, of our college organizing secretary, Dr. K.R. Shanmugam, and conveners, Dr. I.S. Chakrapani, Dr. N. Srinivas, for taking the initiative to organize this seminar. I also extend my thanks to our collaborating institution, Government College, Ramachandrapuram, for their valuable partnership and support in making this event possible. I am confident that the deliberations, presentations, and discussions over the course of this seminar will be highly enriching and thought provoking. I encourage all participants, especially our students and young researchers, to actively engage, ask questions, and make the most of this opportunity to learn from eminent experts in the field. Education is not merely the acquisition of knowledge, but the cultivation of curiosity and critical thinking. Events like this seminar play a crucial role in fostering scientific temperament and encouraging innovation among young minds. Before I conclude, I once again welcome all the delegates and wish the seminar great success. May this gathering lead to meaningful insights, fruitful collaborations, and new directions in biological research. Thank you all.
Thank you, ma'am. And then over to Dr. N. Srinivas from the collaborating institution, GDC Ramachandrapuram.
Very good morning to one and all, and a hearty welcome to this one-day national seminar on frontiers in biological sciences. We are fortunate to have a collaboration with PRR and VS Government Degree College, Vedavaluru, in organizing this seminar. Today we have galaxy of speakers, eminent and versatile in their fields. One Professor M. Rajshekhar Garu from Sri Venkateswara University, Tirupati, Professor L.V. K.S. Bhaskar Garu from Chhattisgarh, and Professor B. Shivkumar Garu from IJR, Tirupati, and Dr. S.B. Sainath Garu from Vikramasinghe University. And the deliberations and the keynote addresses by these speakers will enlighten the participants in various fields and emerging frontiers in biological sciences. We sincerely thank the keynote speakers for accepting our invitation within no time for their valuable keynote addresses. Thank you, sir. Thank you.
Thank you, sir. And over to Dr. K.R. Shanmugam for his initial remarks. Dr. K.R. Shanmugam. So meanwhile, I will be introducing the first resource person, Professor M. Rajasekhar. We are going on an informal note, Rajasekhar.
Yeah, okay.
Happy to share with you all that he is my classmate, he is my mentor, and he is my research supervisor also. Sharing the lineage of Dr. N.V. Nandakumar on academic lines, we have shared the lab and under his guidance, we could produce a good piece of work. And Dr. Rajasekhar is now heading the Department of Zoology, Sri Venkateswara University. And he, for his credit, he has a good number of projects funded by DST and DBT. And he got Young Scientist Award. He is Best Teacher Awardee from Government of Andhra Pradesh. And he has been a member of many scientific and professional bodies and organizations. Also, he is active in other platforms of social service like Vaibhava Bharat. And he is guiding students in cancer biology right now. Right, with a request to start his session. Before that, to give a few minutes to Dr. K.R. Shanmugam for his initial remarks. Over to Dr. K.R. Shanmugam. Meanwhile, Rajasekhar, could you please share your screen?
Okay, right. So is it visible?
Yeah. You can go on with presentation mode.
Right. Is it okay?
Okay, now clearly visible.
Okay. So you want someone to join, or do I need to start?
No, just I am waiting for Dr. K.R. Shanmugam for his initial remarks.
Okay, right. I'll wait.
Okay, then Rajasekhar, would you please proceed?
Okay.
Hmm.
Right. Shall I proceed?
Okay, then.
Okay, right. First of all, very good morning to you all. My friend Dr. Chakrapani and Dr. Srinivasalu sir and other colleagues and my dear friends, all delegates. Very good morning to you all. So I thank all of you for giving me this opportunity, especially the organizers of this event. So my friend asked me to present what I'm doing, but he said all the delegates are faculty members. So I think it's a good thing that I'm going to present my work before all the faculty. So whatever I'm going to tell you, I think you can also have some insights. So the present topic is on TNBC, triple negative breast cancer, understanding the triple negative breast cancer therapies using nanoparticles, which are encapsulated with different kinds of molecules and using them as therapeutic targets. So this is with regard to, just I'd like to share my work before proceeding to what I'm doing, I would like to give a brief note on the cancer statistics, how it is going to affect the population, what are the causes, and what are the therapeutic targets. And then we can go for what kind of research we can do and how we can focus on different therapeutic targets. So if you see the cancer statistics, this is, 15 lakhs, more than 15 lakh new cases are coming out each year in India. So this is the biggest number. Apart from the existing cases, every year we are adding 16 lakhs of new cases. And approximately 1 in 9 Indians are likely to develop cancers in their lifetime. So that is the biggest problem. And of course, 39% of mortality is present among all cancer types. This is because of, these cancers are more prevalent among the aging population due to lifestyle changes. And also, it is more fatal because of improper diagnosis. And that is one thing. And cancer develops over many years, and it is not deadly until very late in its course. Precursor lesions are very important for this, and it can be prevented. Cancer is now more of preventable rather than curing. Curative therapies are very less available. Okay, so that's why we have to focus our full attention on the prevention modes of cancer. And if you see the risk factors and risk reduction, so probably nearly all cancers, like different kinds of cancers, lung cancer, mouth cancer, skin cancer, and bladder cancer, so many types of cancers are there. Among all the cancers, 50 to 75% of deaths are related to personal behavior. So this point we have to note down. Because of our behavior, because of our habitat where we are living, those are also contributing to this much of mortality. And 30% or more of all cancer deaths are related to cigarette smoking. So that means if you see the 50 to 75%, and if you normalize this 75% to 100%, then we can see 30% mortality is related to smoking. And 30% of cancer deaths in US are related to poor nutrition, though it is a developed country. Okay, it's a developed country. The cancer is, attributed to poor nutrition and obesity, inactive and obesity. So this risk may be reduced by increasing consumption of fruits and vegetables that we know pretty well. And nearly 70% of cancers in India are diagnosed at advanced stages. That's why, curative therapy is impossible to restore their health back. So when you see the mortality in terms of incidence or mortality to incidence ratio, relatively, it is very high, indicating late detection. So that is one of the reasons for biggest mortality. And if we see the cancer risk and causes, so there are genetic causes, there are environmental causes, and there are lifestyle causes and personal history. So all these contribute to, cancer. And we see many kinds of things. So this, I told you in the initial stage, cancer can be prevented. So for prevention, what we need to do? So we have to eliminate the infectious diseases. So we have to check, we have to be vigilant on infectious diseases. And also nutrition, what we are taking. Okay, so especially Indian traditional food is healthy. Indian traditional food, again, I'm stressing on the Indian traditional food. Okay, so people are now marching towards the westernization. Okay, our dress code, our food habits, everything is, getting changed to westernization. But if we focus our attention on the nutritive food and Indian traditional food, we can avoid or we can prevent the cancers because, our traditional food, the routine. So we use many kinds of anti-inflammatory materials. Say, for example, curcumin, that is also, having anti-cancer property. And capsicum, that is also having anti-cancer property. Ginger is also having anti-cancer property. And cinnamon is also having anti-cancer property. Okay, and onion is also having a kind of material, a kind of molecule called quercetin, that is also having anti-cancer property. And you name any kind of food material that we are using that is having anti-cancer property. So instead of taking medicine as a food, we can take food, okay, as a medicine, food as a medicine. So then we can definitely prevent the cancer. And the epidemiology of the cancer, probably many of you may be knowing about what is epidemiology. Epidemiology looks for cancer prevention. So it is a kind, it looks for the distribution, determinants, and health-related states or events among population. So that means it looks for the distribution of the particular disease, causatives of the disease, and then it looks for the distribution. And then we can, if we have all such kind of data, we can easily prevent if we know the causes and all. So that is how epidemiology helps for the prevention of the disease. And behavioral medicine. So our behavior itself acts as a medicine. Okay, so what is behavior here? I think you can see this slide. 1 minute, I'll use the pointer option here. Right. So am I audible? Hello?
It's okay, clearly audible.
Okay. So now I think I can show the pointer also. So this behavioral medicine, what is this behavioral medicine? Our behavior itself acts as a medicine. So that means having positive thinking. It plays a vital role in maintaining both mental and physical health. Probably you may be knowing what is health. The definition of the health is the complete physical, mental, and social well-being. Okay, that is the definition for health. And we know that optimistic mind reduces the stress. And further, believing in one's ability to withstand for hard conditions improves resilience and helps the body functions more efficiently. Okay, so that means if somebody do something good, okay, we have to show the gratitude, say thanks, staying hopeful. So whatever that happens, let it happen. Let us, be happy. And for everything, let us be hopeful, for avoiding negative thinkings. That can definitely lower the anxiety and it improves the overall well-being. So that's why, we have to have a kind of positive mindset. It is not just emotional. So it has a measurable benefit on health. So that is why our behavior itself acts as a medicine. And then once the cancer comes, then next step is, if it is in the initial stage, then we can undergo surgery. And there are some cellular and molecular biology targets. Those targets we can focus and then we can prevent the disease. And pharmacological preventions, there are some medicines for early stage cancers so that can prevent the disease. And then imaging, and then public health policy. Okay, so these public health policy is also a kind of public health policy has to be initiated by the government, and the government will take up a kind of epidemiological data, and they have to initiate the program among the public to prevent the cancers or any kind of disease. And again, if you see the causes for cancer, there are two different kinds of cancers, like familial cancer, and the other one is sporadic cancer. Probably you may be knowing about what is familial and what is sporadic. And for these two kinds of cancers, genes are responsible. And the other way, environment is also responsible. Genes may influence to induce the cancer, and in turn, genes will be influenced by the environment. Okay, both together, they may go for familial cancer. Familial cancer means the inherited cancer among generations to generations. So that means from parents to the children, children to their children. So like that, it will be inherited. So that type of cancer is called familial cancer. And these are the genes which are responsible for inherited cancer, like APC, MLH1, SMAD, MSH2. These are all the genes that are responsible for inherited cancers. And there are some sporadic cancers. So those sporadic cancers, cause will be different. It is not inherited. That cause may be, a kind of environmental cause, toxins and radiation. So many causes are there. So those environmental causes, they induce some of the genes like, P53, KRAS, and CTT and BRAF. So like this, these are all the genes which are responsible for sporadic cancer. And there are some common genes like MLH1, P16, MGMT, BAX-T, GFR beta R1. So these kind of genes, they also present in each and every body. They will either come through the inheritance or through the sporadic route. So these are the genetic causes. And then if you see the statistics, what kind of cancer is more prevalent among males and among females, you can see prostate cancer is more prevalent and more prominent among males, and breast cancer is the more prevalent cancer among females. Okay. And if you see the death rate, the prevalence or incidence of disease is more towards prostate and more towards breast cancer among the males and females, and less will be the pancreatic cancer in both the sexes, and remaining will fit into the in between those two. And the estimated deaths will be lung and lung cancers among the males, lung cancers among the females are on the top position. Okay, when compared to deaths. The same thing is being continued for every year. This is statistics taken from reliable sources. And then this picture represents a kind of prevalence and mortalities for different kinds of cancers all over the globe. So this, you can see incidence of stomach cancer, So you can see the entire European region, China, stomach cancers are highly prevalent. Okay, this is South America. So this South America, parts of South America is having stomach cancer. Okay, high incidence of stomach cancer. So like that, in some of the continents, you can find cervical cancer, in some of the continents, stomach cancers, and in some of the continents, lung cancer, okay, and prostate cancer like that. So it is distributed. So that means you can see clearly that there are specific reasons based on the geography. So that means geographically, the incidence or prevalence of the type of cancer is being changed. So that's what we can say. And then you can see the hallmarks of cancer here in this slide. What are these hallmarks? Hallmarks means, so the important key regulators which can induce the cancer, okay, which can induce the cancer. And those indicators will also help as a targets for therapies. So like that, these are all targets. Like, PARP inhibitors, this is a kind of PARP inhibitor, and EGFR inhibitor, cyclin-dependent kinases. So many targets are there. So target means, say for example, some people, they work on apoptosis, programmer cell death. And some people, they focus their attention on the cell cycles. Okay, cell cycle plays major role because, mitosis is the very important segment in cell division. This cancer means uncontrolled cell growth. So that means cell division will not be stopped. As a result, more number of cells will be liberated. So that's a kind of cell growth, uncontrolled cell growth or cell proliferation. So that's why if we stop the mitotic phases or mitotic divisions or cell cycle phases, so we can stop the cancer. That is the version of some of the scientists. And some people, they went deep into the mechanisms and they identified several hallmarks like EGFR, cyclin-dependent kinases, immune-activating anti-CTLAs, telomerase inhibitors, like that. Different kinds of molecular targets they have identified. Among all those, the present slide is showing very important hallmarks of cancer for next generation. And after that, now the present scenario, there are several challenges for treatment with regard to the treatment paradigms. So old paradigm treats all cancer patients with a similar kind of drug. Say, for example, they use, if they identify, if a patient is diagnosed with a specific cancer, like breast cancer, okay, they'll be treated with a similar kind of drug. All patients, irrespective of their gene mutations, irrespective of their molecular mechanisms, they treat only one drug. So there are disadvantages with the treatment. There are advantages also with the treatment. After that, new paradigms entered. So as per the new paradigms, targeted therapy, okay, this is a kind of targeted therapy. So this requires large clinically meaningful studies. So that's why this is a kind of constraint for the researchers to produce therapies. So like this, there are paradigm shift among the therapeutics. And among the identified molecular targets, you can see there are several molecular targets, like targets associated with the specific gene abnormalities, targets associated with cell lineage determinants, and normal immune cells. Okay, and targets associated with different kinds of pathways. Just now I told you, apoptotic pathway, cell cycle pathway, epigenetic pathway. So like that, there are different kinds of pathways. So like that, there are pathways. And then treatment strategies, if you see for the cancer, there are surgeries, radiation therapies, chemotherapies, alkylating agents, anti-metabolites. This is very, very important therapy right now being undergone. So metabolites, there are specific metabolites they are characterizing, and then they are using for therapeutics, and photodynamic therapy, angiogenesis inhibitors, hormone therapies, tyrosine kinases, biological response modifiers. So many treatment strategies are available. And one among such treatment strategies is called nanotherapies. Okay, nanotherapies. So here, what people are doing is any kind of drug can be made into nanoparticles. That is one thing. And then there are some nanoparticles, okay, so those can be used as a therapeutic molecules. Apart from that, there are some liposomal nanoparticles. Say, for example, liposome is a kind of material, okay, lipid material. So we can take that liposomal material, and we can encapsulate our drug molecule. Say for example, this is, this entire thing is a lipid molecule, okay, lipid molecule. We can encapsulate here the drug molecule, and we can make them into nanoparticles. So that becomes liposomal nanoparticle. So that liposomal nanoparticle preparation and synthesis of liposomal nanoparticle itself reduces the toxicity. Okay, so that's why we can use the liposomal nanoparticles. We can synthesize liposomal nanoparticles, and then we can use some of the antibodies. Okay, the tumor-specific antibodies, they can drive the nanoparticles to the target. So that is how we prepare the nanoparticles for treatment, and then we can check the efficacy of those drug molecules. So that means these liposomal nanoparticles and then polymeric nanoparticles, these are also a kind of similar nanoparticles which can control the release of drug material. So that means sustained release drug molecules will be present in polymeric nanoparticles. So these two will help in the drug delivery systems. And after that, there are some therapies, like, we can directly use gold nanoparticles and magnetic nanoparticles, photodynamic nanoparticles, all these nanoparticles directly to the target. Okay, and gold nanoparticles can be used to generate heat. Okay, they can be used directly, and then if as soon as they reach the target, tumor target, and then we can increase the temperature. So that's a kind of thermal killing. Okay, thermal killing of cells. Similarly, magnetic nanoparticles, they also help for thermal killing. So that means they reach the tumor, and once they reach the target, we can use magnetic resonance for creating thermal variations. As a result, the tumor cells will die. So similarly, quantum dots and targeted nanoparticles, many nanoparticles, many kinds of nanoparticles are there. And apart from this, dendrimers, this is also a kind of nanotherapy, nanogels, exosomes, RNA-based nanoparticles, these are all present. And these dendrimers, they help for high drug loading. So that means high amount of drugs can be loaded onto the dendrimers. So that means all these nanoparticles, they help for the improved targeting, and they reduce the side effects, and they enhance the drug efficacy, and early detection plus treatment is possible. Okay, this is called a kind of personalized therapy. Okay, so up to this, I have covered what are the drug targets, what is the prevalence of the disease, and what are the cancers. And now I can just explain what we are doing right now in our lab. So what we do in our lab is called, we do a kind of nanoparticle. Now we are focusing our attention completely on the iron oxide nanoparticles. Iron oxide nanoparticles, because these are very effective nanoparticles in targeting hyperthermia. Just now I told you, hyperthermia means manipulating the temperature, manipulating the tumor environmental temperature to kill the cells or to induce the apoptosis. Okay, so, and that is one thing. And after that, they have low toxicity, because iron nanoparticle, iron is, we need for our body. So that's why it is not toxic. So it is easily biodegradable. So we can, degradable nature is very high. And also for scanning therapies, they are very easy. And apart from that, we take those nanoparticles and we encapsulate nanoparticles with the drug material, and both together we make liposomal nanoparticles. Just now, I told you what is liposomal nanoparticle. We use lipid molecules, we encapsulate the drug molecule along with the iron nanoparticles. Okay, so based on that, we made a particle. Okay, this is the medicine that we are using. So this is a kind of rotenone is a kind of medicine. Medicine means it's a kind of molecule. So this we have taken and we specially synthesized analogs for rotenone. So that means we made some structural changes for rotenone and we synthesized a new drug molecule. Okay, so why we did analog means to reduce the toxicity and also to increase the efficacy of the drug molecule. Okay, so that's how we have taken this rotenone analog and we encapsulated with lipid nanoparticles. Okay, so this is our target. That is how we made a molecule, and we focused on TNBC, triple negative breast cancer. Okay, that's one of the fatal breast cancer types. So we are focusing on triple negative breast cancer. So now what we do, we use this newly synthesized drug molecule and then we test the JC1 expression. Okay, JC1 means that helps to identify the permeability of the mitochondrial membrane. Okay, this mitochondrial membrane, if we see, our drug molecule is inhibiting complex 1 system. Complex 1 system, okay, helps to produce the electron, ETC, electron transport chain. So it is damaging, this medicine is damaging electron transport chain. As a result, electrons leakage happens. As a result of electrons leakage, what happens is ROS, reactive oxygen species, okay, ROS is getting increased. As a result of ROS, increased reactive oxygen species, it induces lipid peroxidation, okay? Lipid peroxidation. So that means lipid peroxidation leads to the damage of the membrane, okay? Membrane. So as a result, cells will die. So that is one route. And the other route, what we have identified, this is a kind of other pathway. So where this complex 1, okay, complex 1, which is being inhibited, and this inhibition will suppress the UHRF1. Okay, UHRF1. This UHRF1 is also called CBP90. This is a kind of epigenetic regulator protein that plays a major role in DNA methylation. Okay, so this is epigenetic regulator protein that plays major role in DNA methylation. So that means it it helps for chromatin remodeling and gene silencing. So this is frequently overexpressed in many cancers. This UHRF1 is overexpressed in many cancers, and it contributes to the tumor development. So that's why what we did is, we have observed the UHRF1 expression. As a result, this is suppressing. So that means down-regulating this UHRF1, okay? Down-regulating. So as a result of down-regulation of this UHRF1, this is a precursor protein for DNMTs, DNMT1, DNMT3A, DNMT3B. These DNMTs are also called as DNA methyl transferases. Okay, so these DNA methyl transferases, they are very actively involved in epigenetic mechanisms. Epigenetic mechanisms. Those epigenetic mechanisms, what is epigenetic mechanism? Probably may be knowing. Epigenetic mechanism means DNA methylation changes. So this UHRF1 helps maintaining epigenetic information during cell division. Okay, so that means it recruits DNA methyl transferases. So if it is present, okay, if it is present, it regulates DNA methyl transferases. So that's why, it is getting down-regulated with the drug. As a result, these are DNA methyl transferases are getting activated to, repair the damaged DNA. So as a result, BAX genes, BIX genes, cytochrome C, caspases, these DNA methyl transferases are influencing all these BAX, cytochrome C, and caspases. As a result, it is leading to apoptosis. Okay, apoptosis means it's a kind of programmed cell death where cells are being dead. So that is one thing. And this is, very little deep into the mechanisms. We just went. So you can see here, when we see the DNA methylation changes, so here you can see DNA methylation means adding of methyl groups. This is CH3 group. Okay, this is cytosine region, and this is guanine region. So you can see this methylation changes will happen cytosine followed by guanine. Okay, so that is called, hypermethylation or hypomethylation. Hypomethylation means if it is less, methylation changes are very less than hypomethylation. If it is very high, hypermethylation. But those will be present in between cytosine followed by guanine. So we call them as islands, CPG. So CPG islands, we call them CPG islands. And we identified those CPG islands where they are hypermethylated and where they are hypomethylated. So we identified more hypomethylation than hypermethylation with our drug. So that itself indicates the drug is working very fine. Okay, so that is one thing we have identified. So that means methylation, this methylation, hypermethylation especially, silences gene expression by preventing transcription. Okay, so this hypermethylation helps to, silence the cancer promoting genes. So that is one thing. And after that, so this is clearly evident that, the pathways are being disturbed, cancer pathways are being disturbed, so apoptotic mechanisms are being promoted. So that's one thing. And after that, we have seen some of the calcium pathways, okay? So this complex 1 increased ROS helps to increased pathway, increased ROS helps to H2O2. So there's a kind of free radical formations. They help calcium mobilization. Okay, calcium mobilization from intercellular region to intracellular region, intercellular region to intracellular region, where an enzyme called calmodulin. Okay, this calmodulin enzyme will help for the calcium pumping mechanisms. So this calcium is also being influenced with rotenone encapsulated drug molecules. So that is one key finding that we have identified. And after that, what is this ferroptosis? So I told you we are using some iron nanoparticles, okay? Iron nanoparticles. These iron nanoparticles, you can see, they target on glutathione, GSH, okay? GSH. So this GSH induces as a result of oxidation, okay? GSH as a result of oxidation, it will be converted to glutathione peroxidase. This glutathione peroxidase, the number, if the increased amount of peroxidase is present, it induces lipid peroxidation, okay? As a result of lipid peroxidation, ROS will be generated. Okay, ROS will be generated. This ROS will kill the polyunsaturated fatty acids, PUFAs, polyunsaturated fatty acids. So this leads to the cell death. That is one more mechanism. So that means ferrous-induced apoptosis, we can say. Ferrous-induced apoptosis called ferroptosis. Apoptosis is different and ferroptosis is different. Apoptosis is a kind of natural mechanism or that can be induced through the drugs, whereas here we are using ferrous molecules, iron molecules for inducing apoptosis to kill the cancer molecules. Okay, so this happens only with the cancer molecules, not with the healthy cells, because there is another mechanism that plays a crucial role. So that's one thing. And finally, we have checked the cell cycle pathways. So here, so many parameters are here. So like probably you may be knowing, all of you are science teachers. So probably what is a cell cycle, you may be knowing pretty well. So there are M phase, G1 phase, G2 phase. So like that S phase, 3 different phases are there. So all these phases are having a kind of cellular switches. Those switches are called cyclins. Cyclin D, CDK4, okay. Cyclin B1, CDK1, P21, cyclin E, CDK2, CDK2. So these CDKs and cyclins, they regulate the cell cycle mechanisms, okay? And we have seen using this rotenone encapsulated nanoparticles where exactly cell cycle is being stopped, okay? That we have identified, that I'll show you. So that means we have checked this cyclins, we have checked this CDKs, okay, and cell cycle assays also. So before, so these are all, the pathways we are looking onto the cancer therapeutics. And now, what kind of experiments we did, I'll show you. So this cytotoxicity analysis. So initially, we have cultured the cells called MDA-MB231 and MDA-MB365. So those 2 cells we have cultured, and we did a kind of cytotoxicity analysis by using the drug material what we are using with the different concentrations, like 1 micromolar per ml concentration, 2, 4, 8, and 10 micromolar concentrations. Surprisingly, these molecules are working with lowest concentrations on par with the cancer drug that is available right now. So we have checked with the cancer drug molecule, that is staurosporin. We have used staurosporin as the control, and it is working on par with staurosporin. So that is a wonderful thing that we observed. And this MTT assay shows the cytotoxicity. And after that, we have seen JC1 expression. Okay, this JC1 expression helps to identify the mitochondria membrane potential. So because of changed mitochondrial potential, we can see the absorbance will be different. Okay, radio labeled tags we use. Based on that, we check the membrane potential, and then we can see whether that leads to apoptosis or not. And again, we have seen the apoptotic assays. So this apoptotic assay For this, we have used annexin 5, FITC, and propydium iodide staining using flow cytometer. We have checked the apoptotic mechanisms. So with the lowest concentrations, the cells are being killed. And this is a kind of, TEM pictures with nanoparticles for checking iron nanoparticles, whether they are encapsulated with the drug molecule or not, that we did. And these are the cells you can see, okay? This plate, so this is, 2 micromolar concentration. And this is, you can see the agglutination of the cells. So that means cells are dead. Some of the cells are dead. And this plate shows confluency among the cells. Confluency means full plate of cells. You can find full plate of cells. So based on that, we check the MTT assay for getting ideal concentrations. So this is JC1 expression. So, these indicate how the membrane potential is being changed, right? With control and experimental cells, in vitro cell lines. We used in vitro cell lines. So you can find here, this is the kind of apoptosis. So here MDA MB cell 231 and MDA MB 231. So this is the other one, okay? So this is the other one. This is a kind of cell cycle arrest. So here you can find, this picture shows, a kind of signal. Whereas here, complete shift of cells from here to here you can observe, right? So this indicates shift of cells from one phase to other phase. So that means, so here this is untreated cells. These are treated cells. These are other kind of treated cells. So you can find, completely switch of cells from one phase to other phase. So that means their cell cycle is being arrested. So here you can find 37.3% distributed in Z0 and Z1 and 7.4% in Z2 and M phase. After treatment, it was found that there was an increase in Z0 and Z1 population, that is 46.5%, with concomitant decrease in the S phase population. So that means S phase is being arrested. Okay, so that's how, we have seen these things. So that means now it is clear that cell cycle is being arrested and apoptotic mechanism is also being initiated. Okay, and membrane potential is being changed. Mitochondrial membrane potential is being changed. After that, we wanted to look for what kind of specific genes are responsible. Okay, what kind of genes are overexpressed. So just to have genetic signatures, we did a kind of transcriptomic analysis. Okay, so based on that transcriptomic analysis, we have identified 81 to 83 genes, okay, which are down-regulated, which are up-regulated. Okay, so more than 100 genes were identified in treated cells, in treated combinations. So these are, some of the volcanic plots showing distribution of differentially expressed genes across the two pairwise comparisons. So these are some of the heat maps we have identified for differentially expressed genes. Apart from that, we identified 10 differentially expressed genes among untreated to treated. So that is our conclusion. And after that, one specific gene, that is a kind of hub gene, has been identified. So that means that hub, is transmitting or regulating all the parameters. Okay, so that has been identified. That hub gene has been identified. So based on this, this up to now, what we did is only in vitro studies. Only in vitro studies we did. Okay, now we wanted to go for in vivo studies based on all these parameters, and the presence of miRNAs are also helping the treatment. Small non-coding RNAs, that is microRNAs, which are present in JAS treated samples, and total 22 miRNAs were identified in the treated samples. Okay, so this is also a kind of indication that these drugs are getting worked. So this is how, we did for several assays. Okay, this is what we are doing right now and what we are going to do from hereafter. So that is working on in vivo models. So finally, I just would like to conclude because time is up. Prevention, is better than cure. We know pretty well prevention modalities. So this prevention is not available in stores, okay, and drugstores. So to purchase. So how much is an ounce of prevention? That is not available. So that we have to do. So that means we have to change our attitude. We have to change our behavior. We have to change our habits, like food habits and mobility. So everything we have to do. I told you one thing here called, thermal regulations, body hypothermia or hyperthermia. So that means temperature changes, exercise. These are also helping to reduce the cancer burden. So that's how we can march towards prevention of the cancer. Okay. So finally, I thank the HRD DHR for the grant support and also DST for supporting me and my students, Mr. B. Lokesh Reddy, Pratima Bharati, Manikyam. Okay. And I should not forget my better half, Dr. G. Lata, for supporting me for the entire path of my research and all, for things. Thank you very much for giving me this opportunity. If you have any questions, you're welcome. So any questions?
So now, thank you, Professor Rajsekhar. Over to Dr. Shanmugam.
Good morning, sir. First of all, I thank the Professor Rajsekhar sir for valuable and scientific information, sir, about the cancer biology. And we have learned so much of information about the cancer biology, sir. Thank you, sir. Thank you, sir, for your nice presentation, sir. Thank you, sir.
Thank you very much. And I would like to say one thing here. Very recently, we have received ANRF pair grant. Okay, there is a concept called ANRF hub and pair. Sorry, no, it's not hub and spoke concept. Okay, so under which SV University got 15 crores of grant. Okay, so this is for entire hub. Central University of Hyderabad is acting like a hub. Under Central University of Hyderabad, 7 universities are acting like spokes. All hub and spoke together got 100 crores grant from ANRF. So from which 15 crores is SV University share. And I'm happy to say that I'm also one among the PIs from SV University. So I got 1.5 crores to working on these lines. So with which I have established a clean room facility that is a kind of state-of-art cell culture laboratory. Okay, that is available, probably that those kind of labs are available in central institutes. That facility is right now available in our university. So that's, a proud moment to share with you.
Congrats. And then any questions from the participants?
Any questions? If you have, you are welcome.
Then one doubt expressed on message. Tumor microenvironment. Adokar చెప్పమంటున్నారండి.
Tumor?
Microenvironment.
Tumor microenvironment is a big word. Microenvironment means there are so many factors which are present in and around the tumor. Because, glycolysis pathway will also contribute. So that the tumor will attract more amount of sugars. It won't allow any cell to take sugar component. It takes the entire sugar from the environment and it grows. Similarly, the blood vessels, angiogenesis is also one of the factors. More number of blood vessels will be formed to supply more amount of oxygen towards the cells. So, this kind of things are playing crucial role in microenvironment. So if you are specific about a question, I'll answer.
Yeah.
That's a big task, microenvironment.
And then one question about dendrimers. అంటే dendrimers అంటే ఏంటి అని అడుగుతున్నారు.
Dendrimers.
Somewhere crept in between your lecture.
Yeah, dendrimer is a kind of nanoparticle.
Oh,
Yeah, I have shown you. So dendrimers, it's a kind of, synthetic molecule that, a kind of three-dimensional structure widely used in nanotechnology for biomedical applications. That dendrimer can be used for encapsulation of the drug molecule, like lipid molecules. I told you, okay? Initially lipid molecules, pegylated molecules, PEG molecules, okay? So those are the things. That's a kind of, synthetic molecule with which we can encapsulate the known drug molecules.
Okay. So anything more from the participant side?
Good morning, sir.
Good morning, madam.
Hello. Good morning.
Good morning, madam. Tell me.
I'm Ankamma. Glad to meet you, sir.
Madam. Thank you. Good morning.
Okay. Nice lecture, but I'm sorry I have joined after 10:30, 10:40. I didn't get late, right? I want to have one clarification, sir.
Okay.
How can we correlate thermoregulation and cancer biology? Because I didn't clearly got the point.
Yeah,
So,
here the thing
have
is
clarification about that.
hyperthermia, madam. I used a term called hyperthermia.
Okay.
Hyperthermia. So
Okay.
hyperthermia is a condition where in a micro environment of the tumor, okay, we can focus our attention to increase the temperature in the micro environment.
Okay.
So like some of the particles, magnetic nanoparticles we can use, we can increase the temperature based on a mechanism, especially in the tumor environment. As a result of increased temperature, the cells will die. They initiate apoptosis.
That is through medication or by regular exercise increasing the body temperature?
Yeah. This, by regular exercise, the body gets warm up. That also helps.
Okay, sir. Okay. Thank you very much and nice lecture, sir. Okay.
Okay, then Rajasekhar, our friend Professor LVKS is also online.
Okay.
Bhaskar sir, welcome.
Sir, namaskar, sir.
Bhaskar, how are you?
Good morning. Namaskar. I am good, sir.
Okay, right. I'm happy to see you after a long time.
Hello.
Over to Dr. N. Srinivas. So if is there anything more to speak with Professor Rajasekhar from the participant side? Gurumurthy,
yes, sir. Yes, sir. So good morning to all. Good morning, sir. Rajasekhar, good morning.
Good morning, sir.
Good morning to LVKS Bhaskar also.
Morning, sir. Gurumurthy, sir. After a long time.
Good morning. How are you, Bhaskar?
I'm good, sir.
Right. Thank you. I have a question for Rajasekhar, sir.
Yeah, please tell me, sir.
You have explained very well on cancer. You told about apoptosis and another word. What is that? I didn't get it.
Ferroptosis.
What is that meaning?
Apoptosis induced by ferrous molecules like iron nanoparticles.
Yes. Okay. Okay. Thank you. Sir. Okay. Carry on. Sir. Okay. Thank you.
Okay, then thank you, Professor Rajasekhar, on behalf of the two institutions, collaborating institutions, and the participants.
Right. Okay. Anyway, dear Chakrapani, I'm taking leave because I need to go for the department.
Okay, I understand.
I'll take leave.
Okay.
Thank you very much for giving me this opportunity.
Thank you, sir. Thank you so much, sir. Thank you.
Okay, sir. Thank you. Bhaskar, I'll see you again.
Yes, sir.
Okay, bye.
Okay, now over to Dr. N. Srinivas.
Welcome, sir. LVKS Bhaskar Garu. Hearty welcome. Now I take the privilege to introduce one of the keynote resource persons today, Dr. LVKS Bhaskar Garu, working as Professor, Department of Zoology, Guru Ghasidas Viswavidyalaya, Bilaspur. His professional experience includes he worked as Director, Research and Development Cell, Guru Ghasidas Viswavidyalaya, Bilaspur, and Director, IQAC. From 2014 to 2019, he worked as Senior Scientist, Sickle Cell Institute, Chhattisgarh, Raipur. From 2012 to 2014, He worked as associate professor, Sri Ramchandra University, Chennai, from 2012 to 2014. He worked as associate professor again in Sri Ramchandra University, Chennai. From 2003 to 2007, he worked as postdoctoral fellow in CCMB, Hyderabad. His research grants as PI, principal investigator, includes in 2021, he got a research grant from Association of Applying with Vasoaccular pains, crisis and pain measures in sickle cell disease. ICMR ongoing project is 25 lakhs, worth of 25 lakhs. Again in 2021, he got evaluating the interrelationship between miRNA expression SNPS in miRNA target genes in sickle cell disease. That is CSR funding, worth of 32 lakhs. In 2009, he got identification of susceptibility genes associated with non-syndromic cleft lip with or without cleft plate in Indian population. Again, this is funded by ICMR, and this project is completed, worth of 25 lakhs. And 2010, he got another project, evaluation of insulin metabolic genes polymorphisms in pathogenesis of polycystic ovary syndromes, PCOS, funded by DBT, and it is completed with a worth of 21 lakhs. And 2011, he got another project, that is the role of microRNA in metabolic syndrome, from CII, Indo-Taiwan Collaborative funded project, and it is completed. And he visited Taiwan under this project. And 2015, he got another project, that is identification of correlation of beta globulin gene cluster haplotypes with the clinical and hematologic features of the sickle cell anemia in Chhattisgarh Council of Scientific and Technology, completed with a worth of 5 lakhs. And he guided 5 PhD students and 25 master dissertations. And publications, he has a total number of 248 publications with 4,153 citations, having H index of 29 and I-10 index of 101. And he has the membership in various scientific organizations, which include life member of Ethological Society of India, ESI. He is life member, Indian Society of Human Genetics, and the governing body member of Indian Society of Human Genetics from 2011 to 13 and 2015 to till date. He has presented nearly 40 papers within India and visited abroad and presented 3 international papers in Australia, Taiwan, and USA. To his credit, he has best teacher awards, 3 best teacher awards from his own parent university, and best presentation award, that is Professor K. Pampapadirao Memorial Gold Medal for Best Young Scientist Presentation 2003. He is an eminent scientist in genetics, and now we have fortunate enough to have his talk today. He is going to present his talk on genetics study designs. I request Professor LVKS Bhaskar Garu to address the audience participants over this seminar. Over to Bhaskar Garu.
Thank you very much, sir. Thank you very much for the elaborate introduction. At the outset, I would like to thank the organizers, especially our senior professor I.S. Chakrapani sir, for giving me this opportunity. Next 45 minutes to 1 hour, I'm going to talk on genetic study designs. Maybe it's not a research related topic, but maybe if anybody wants to design some studies in future, maybe this will be helpful for you. And understanding this topic, you may design some of the studies in genetics in your field, in your area.
Bhaskar, 1 minute, please.
Yes, sir.
ఆడియన్స్ లో కొంతమంది హ్యుమానిటీస్ ఫ్యాకల్టీ కూడా ఉన్నారు.
ఓకే సార్.
సో కొంచెం జనరలైజ్ చేస్తే కూడా బాగుంటుంది.
ఓకే సార్.
థ్యాంక్ యూ.
Thank you, sir. I will just try to have some generalized topic, but okay, sir. I will try, sir. Thank you. I hope that my slides are transition, sir. Moving,
Yes,
sir?
sir.
Yes, sir.
Yes, sir.
Genetic study designs. So everybody knows that people or animals or anything will be having the diseases. So disease means any deviation from the normal health condition. So some are age-related disorders, some are gender-related disorders, some will be having the genetic basis, and some will be having the environmental components, and some will be having the infectious agents involvement or chemicals involvement. So here any disease means that disease will be having exclusive genetic component, exclusive environment component will not be there. And genetic disease is the culmination of both genetic factors and environmental factors. The interaction of several genetic factors or interaction between the genetics and environmental factors is leading to the disease. So some diseases are genetic diseases we say, or heritable diseases we say. When we talk about the heritability is very much important because how a genetic disease, a disease will be inherited from the parents to the children. So that means the basic question, for example, any disease, if you say that, okay, whether the disease is heritable or not. So for example, if you say the diabetic, okay, anybody in your family, your mother or father is having the diabetic, this type of questions we ask. That means whether this disease is having any heritable component. So heritability is the proportion of the phenotypic variation in a trait that is accounted by the genetic variation. For example, 30% of the genetic basis is there. That means 30% of the genes are responsible. Remaining 70% may be having the environmental component. So then what proportion of the phenotypic variance is contributed by the genetics or the genotypes or any genetic components or the chromosomal component or anything. So heritability can be estimated by comparing the correlation between the family members. So in a family, you can just see the several individuals may be having the disease and their relationships also. For example, if you take the 4 or 5 generations of the family or 3 or 4 generations of the family, if you take it, how the family members are having the disease and what is their relationships and what is the component of the genetic variation is involved in that particular or observed in that particular cohort of this one. So heritability is H square is equals to changes in the genetic component by the changes in the phenotypic component. That means sigma genetic component and also sigma phenotypic component. We just say that this is the equation what we generally use it. So for identifying the heritability, for example, if you say that alcohol drinking is a heritable component, if father is drinking or the mother is drinking, and that will be giving to the children. So when we talk about the genetic component, if you want to identify the genetic component involved in a disease, can be identified by estimating the heritability. This heritability will be generally estimated using the twin studies. So generally some of the institutions will have their own twin registries. For example, very famous of Australian twin registry is there. Australian people, they just recruit the twins and they use for identifying the genetic component in the disease. So here VP, that means variations in the phenotype is culmination of genetic variance, environment contribution, and also other environment contribution that is called unique environment contribution. VP is equal to A plus C plus E. A is equal to genetic variance. What is the genes? What are the genes that a person is having? And what is the common environment? What is everybody is having that environment? That is a common environment. Unique environment. That means in the mother's womb, what kind of environment the person will be having. So twins, if you see, you can just see the two kinds of twins, monozygotic twins or the dizygotic twins. Monozygotic twins means 100% genetic environment will be there. They are common, having A is 100%, and dizygotic twins, you can find only A is only 50% because they develop from the two different embryos. So embryonic environment is entirely different. So the dizygotic twins, they have 50% of the genetic environment. Monozygotic twins, they will be sharing 100% of the genetic environment. So monozygotic and dizygotic twins correlate 100% for the common environment. So they have developed from the same uterus, they will be developing. And monozygotic and dizygotic correlate for the exclusive environment. There is no exclusive environment for these people. They will be having the 100% common environment. These are the different notations you should understand for that. Here, just a very simple calculation. I'm just showing that if you understand, it's fine. If you're not understanding also, you need not worry about it. A is equals to, that is the genetic component. C is the common environment. E is the unique environment. So A is equals to 2 into correlation of monozygotic twins minus correlation of dizygotic twins. C is equals to correlation of monozygotic twins minus genetic component. E is equals to 1 minus common environment, or the monozygotic correlation of the monozygotic environments. So if the values you just see this here, correlation of monozygotic is 0.70 and correlation of dizygotic 0.40. Then you can just calculate and you can get note that the gene environment interactions increases with the E. As long as this unique environment is increases, so gene environment interaction also will be increasing. So unique environment increases, gene environment interactions will be increasing with this particular thing. So you can assess that how much is the common environment is involved, how much is the genetic. So for example, in this scenario, 60% of the genetic component, 10% of the common environment, and 30% of the unique environment is involved in this particular disease. So what are the genetic diseases? We say that diabetes is a genetic disease. Sickle cell is a genetic disease. Maybe cystic fibrosis is a genetic disease. Maybe you can just say PCOS is a genetic disease. Genetic disease means they cluster in a families. So you can find the multiple members in a family will be affected. So very clear pattern of inheritance may be there and may not be there. For example, diabetes, you see this. So you cannot say that whether mother or father is having the disease that will be coming to the children or not. We don't. How many children will be getting, whether autosomal dominant fashion, autosomal recessive fashion will be followed or not. Nobody knows that. So, but it clusters in the families. See, multiple diabetic members will be there in the families because that is a genetic disease. PCOS is there. PCOS also multiple members may be there. So the identification of the disease gene is very crucial for the genetic disease, if we say. So genetic diseases identification, gene identification is very helpful for the pathogenesis mechanism. For example, alcoholism, you say that alcoholism is taking the alcohol is a disease. So we talk about the ADH. ADH, very fast gene is there, then that people will not become the alcoholics. For example, East Asians. East Asians will be having the very fast ADH gene. Very slow ADH gene is present in our Indians because once we started drinking, we become addicted. So that's why as long as you are not started the drinking of alcohol, you will never become an alcoholic. Once you started drinking the alcohol, then that understand that maybe pathogenesis, pathogenic mechanism underlying the disease can be identified if you know the gene. And also development of diagnostic by identifying the mutation, maybe you can just say that the person is having the disease or not, maybe we can just say. So multiple individuals is, and some will be having the carriers and some will be having the disease. So genetic diseases can be denoted in the two different things because one is the simple Mendelian genetics or the simple genetic diseases, and second one is the complex genetic diseases. So simple Mendelian genetics, we talk of, for example, sickle cell disease, Huntington disease, cystic fibrosis, muscular dystrophy. These things will be having a very clear inheritance pattern. If both mother and father is having, and children may be having definitely. Diabetes is not a simple Mendelian genetic disorder. So if mother is having, maybe children may get the disease or may not get the disease. But sickle cell allele is there in the parents, then definitely it will be contributed to this one. Huntington disease also is like that only. Cystic fibrosis. So that here complete correlation of the genotype. If the gene is there, defective gene is there, disease will be there. You get a mutant gene, you will get the disease. If you don't have the mutant gene, you will not get the disease. This is a very simple Mendelian genetic disease. Here the Mendelian diseases are like thalassemia, sickle cell anemia, cystic fibrosis, hemophilia, these kind of things are. So complex or multifactorial disease. Complex disease is very hard to understand. Diabetes. So nobody knows that what gene is exactly causing the disease and what kind of inheritance pattern. My mother and father is having diabetes and whether I will be getting the diabetes or not. So nobody knows that. Very hard to analyze. And many common diseases will be associated with this particular, comes under this particular complex multifactorial disease. For example, cancer. So cancer, no single gene is responsible. Asthma, no single gene is responsible. Schizophrenia or any other mental disorders, hypertension, heart diseases, diabetes. These are all will not be having a single etiological factor or the single genetic factor. So the risk of getting the disease is modified by the individual genotype. So for example, I am having one defective genotype. So what are the other habits? So whether I am the binge eater, or otherwise I am having very other smoking or alcohol related habits. So these things will be worsening the particular person may be having the genotype. When the person is having the other risk factors or other environmental factors, the person's chance of getting the disease will be higher. Other risk factors, especially the other genes. So not only single gene will be caused, whereas in this particular disease, so sickle cell anemia is a single gene disorder. So one particular gene, that is HBB gene, 6th amino acid, if glutamate is replaced by the glutamic acid is replaced by the valine, the disease will occur. Here it is not like that. One single gene is not causing the disease. Many of the genes and many of the environmental factors, gene to gene interactions, gene to environment interactions, lifestyle parameters, all these things will be leading to the disease. So disease penetration also will be different. Because what extent, for example, some people will be needing, for example, diabetes you take. Diabetes, some people will be dependent on insulin. Some people, they never require any tablet, little lifestyle modification with a little reduced eating habits and also very less walking. This type of things, other lifestyle parameters, they can just manage and they never take that medicines or any other things. But some people will be needing 3 or 4 groups of medicine. Some people will be needing insulin. So these are the different parameters. You can just see the complex disorders. Very hard to analyze these particular disorders. So whatever it is, identifying the gene is very much important. We have 23 pairs of chromosomes and 22 pairs is called as autosomes. One pair is called as a sex chromosome, and the female will be having the XX chromosome, male will be having the XY chromosome, and it's a very large genome. They contain thousands of the genes. Earlier we were thinking that more than 2 lakhs genes were there, and one gene, one protein concept, people are thinking more than 2 lakhs genes are there. Nowadays, now the gene number is reduced to 25 to 30,000 genes, because still we are not very clear that how many genes are there. We think that now 25 to 30,000 genes are responsible, and finding a particular gene responsible for a given phenotype. Phenotype, I talk for the only disease. Disease means phenotype. So for example, phenotype means many things. For example, height of the individual, weight of the individual, or color of the individual, hair length of the individual, hair color of the. These are all phenotypes. I'm not talking about, I'm just talking about the diseases we are talking. A particular gene responsible for a particular disease is identification is very difficult, because we have 20,000 genes or 25,000 genes. So identifying the gene is a very big, this one, it is almost equivalent to the identifying the nail that is fallen in the grass haystack. And the finding the genes, initially genes were identified by the pure position independent methods. So they don't know the position. Where is the chromosomes? What are the different chromosomes? What are the different polymorphisms in the chromosomes? We have 23 pair chromosomes, we know that. So other than that, genetic information and gene mapping information, we don't know. And also we don't know the techniques. We don't know the PCR. We don't know the DNA sequencing methodologies. We don't know the Western blotting. We don't know the Southern blotting. No techniques were available before 1980s, I am talking. So at the time, we were identifying the genes. We have identified the genes, that is only pure position independent methods. So that is, I will come to the next slide. What is the position independent method? If the biochemical basis of the any disease is known, we can identify the disease. So biochemical basis of the disease is known. For example, clotting factors, blood clotting. For example, if you have any cut in the body, immediately after some time, the blood leakage will be stopped. That is because of the clotting. If the clotting is proper, then it will be stopped and you will not be having any problem. So this clotting will be facilitated by the factor 8. And so factor 8, any problem is that they will be having the clotting disorders, anything related to the, because they will be losing continuous blood or loss something, maybe during surgery, they will be facing the problem. So it may be possible to purify and partially characterize. For example, if you know that clotting factor is responsible for the clotting related diseases, maybe you can identify the protein that is responsible for the clotting, and gene specific oligonucleotides or the specific antibodies can be generated, and these things will be used for identifying the genes. Gene specific oligonucleotide. Oligonucleotides is nothing but the AGCT. Oligonucleotide is nothing but the adenine, guanine, cytosine, and thymine. These things one can identify. So this picture shows you about how this factor 8 was identified. People identified. So anybody is having the hemophilia, that means blood clotting, blood is not clotting, that's called as hemophilia. So here factor 8 is identified. This is the gene responsible for that. What they identified after that, they first deduced the amino acid sequence. They identified the amino acid sequence. So after getting the amino acid, reverse translation method, they just get it. Each amino acid, one nucleotide codon will be there, and they make that particular codon and then synthesize the oligonucleotide library and the probe library they generate, and they just get the structure of the gene by hybridizing with that particular clones. So they identified the gene in this particular manner. This is called as the position independent method. We don't know the position, we know the protein, and the protein was reverse translated, and from that we got the DNA sequence. From that DNA sequence, we have identified the gene that is responsible for the clotting is factor 8. So there are some revolutionary things that occurred over a period of time. And in 1975, Edward Southern has identified the Southern hybridization, and Sanger in 1975 identified the DNA sequencing methodology. David Boyston 1978 identified the RFLP, that is the restriction fragment length polymorphism. Using some enzymes, you can cut the DNA, and using some enzymes, you can just attach 2 DNA copies. So that's called RFLP is dividing the DNA into the 2 pieces or cutting the DNA at certain places. In 1983, Carey Mullis identified the PCR, and this has and 1989 microarray. Microarray means expression profiling of many genes in a single step, that is given by the Stephen A. Fodor. So these are the techniques that has changed the molecular biology, and now present day genome sequence has been identified and complete sequence mapping has been done. So announcement of first draft of gene was 2000, because this gene draft sequence was published by 2003. Entire sequence of the human genome has been delineated and it is completely annotated, and major journals it has been published in 2003, Nature, Science published. So they have just, it is a very revolutionary change in the molecular biology. So before that, what happened? So many techniques, PCR, RFLP, Southern blotting, and all other techniques has come into existence. Now we know that how to cut the DNA, how to sequence the DNA, all the things we know. And by and large, we know the sequence. So the sequence is characterized, sequence is annotated. So sequence is having so many changes. So you and me is having, for example, in this particular platform, we are having 30 to 35 members. If you analyze the sequence, all individual sequence is entirely different. More than 99.99% we are similar. So this 0.001% is varying from you and me. So that is that variations has been delineated and has been identified. So some are tandem repeats, they are repeatedly present in this. Some are deletion, some are insertions, some are alu repeats, some are repeat expansion, some are long nucleated elements, some are transitions, simple transition, A to T or A to Z or C to T, and transversions and deletions, insertions. So these things has been identified. For example, you can just see that CT repeats, or ATC repeats, ATCT repeats. So like that, different small repeats. These are called as the micro satellites or mini satellites kind of sequences. So we have identified so much of variation. These are the different variations that has been identified in the human genome. We know the sequence, we have techniques, we have polymorphisms. that are variations present in the genome. So these things are available now. Now we need not go for any of the position independent methods to identify. For example, so when factor 8 was identified, we don't have this technique. That's why we have done many things. Now we have almost all the techniques and human genome variations. You just see this. So non-synonymous substitutions can change the amino acid. One nucleotide change will change the amino acid and entire protein will be changed. Sometimes this particular change of the nucleotide may create some stop codons. So protein, instead of 150 basis protein, only 50 base protein only will be formed. That is protein truncation, premature truncation, premature stop codons will be formed. Insertion deletions are there. If anything suddenly inserted, what happens? Entire frame will be changed. Or if anything is deleted, then entire sequence will be changed. So these are the different things can be seen. And mutations may affect the splice sites. Alternative splicing and different proteins from one single RNA will be developed. So these things will be, you can just see that these are all the things are contributing to the functional variations in the protein, or many of the proteins are deleterious proteins, very good proteins. These things will be formed. So these variations contribute to the functional variation in an individual. So these are all with this. So this particular thing is showing that 2010 Nature paper and 1000 Genome Project. After analyzing that, human beings are having 60,157 synonymous protein. Synonymous SNP means it's a single nucleotide variation. Only one nucleotide variation will be there. And these things will not be changing the amino acid. Mutation will be there. Because of this mutation, no change in the SN codon, no change in the amino acid. So non-synonymous means if mutation is there, mutations, because of the mutation, you can just see the variation in the amino acid. So like that small in-frame mutation 714, stop losses 77, and like that genes disrupted by the large deletions. Because of 147 large deletions, the gene is disrupted. Gene is disrupted means protein will not be formed. So protein is not forming means maybe some disease will occur. So these are the different nucleotide variations has been identified. Now you can just see this. So variations based on the frequency, we just talk about. For example, people talk about what is the mutation and what is the polymorphism. People generally ask this type of questions. So polymorphism is present in the many population. Its frequency will be generally more than 5%. And mutation means mutation is exclusively present in the disease conditions and very less frequency will be there. And here the graph is showing this. So here graph, you can just find the x-axis allele frequency and y-axis you can just see the effect. So very low effect in the bottom, very high effect on the top, and very low in the corner side, x-axis, and very high common in this one. The mutations frequency is very less, it is present here. Very high frequency, it is present here, and effect size is more. So that means that mutation is causing major changes, then it will be shown here. Very less changes, it is shown here. So based on this, you can just see the picture. And rare variants, very less frequencies. When 60,000 individuals, one individual will be having the mutation. Very rare. You cannot find, for example, lysosomal disorders. They are present only in 1 lakh individuals, one individual will be having this. This is a rare variant that is called, because they will be having the very small effect in this one. And sometimes rare alleles also causing the Mendelian diseases. Rare alleles, maybe for example, sickle cell anemia, you take it. Sickle cell anemia, it's a very rare in our Andhra Pradesh, you cannot find the sickle cell anemia. It is present only in Kondadora, some populations in tribal. Only one tribal population is having the very rare allele. That is causing the Mendelian disorder. And whereas in Chhattisgarh, it is not a rare allele. It is very common allele here. And it is causing the disease. Sickle cell anemia means it is present, it is a blood-related disorder. If the person is having the mutation, that person's hemoglobin will be changed into the sickle shape. So generally RBC will be donut shape, very round shape will be there with beautiful red color RBC will be there. Once the deoxygenated condition, blood oxygen levels is depleted, then person's hemoglobin will become, RBC will become as a sickle shape. Because of this sickle shape, they call as a sickle cell anemia, we say. Because of the disease, the person will be having the anemia because this blood cell will be very quickly dying when compared to the normal hemoglobin. Normal person's RBC survive approximately 120 days. whereas sickle cell anemia patients, approximately it survives only 10 to 20 days. So like that, because the rare allele may cause very small effect or maybe very high effects. At the same time, very high frequency it is there. It is a very common variant. This common variant is implicated in the very low effect. For example, it's showing low effect, or it may show that high effect also. If it is showing the low effect, then that identification is very difficult, because you have to identify using the GWAS study. GWAS study means, so entire genome will be analyzed to identify the gene. That is called as a genome-wide association study. So whereas here, if the common variant is causing the disease, common disease, then maybe you can just say that its influence is very high in the population. So this is just to show that one. And low frequency or the moderate frequency is there, and moderate, immediate, intermediate effect it will be shown. So that means it is rare variant or the common variant or the moderate variant. They may cause either extreme conditions or extreme high effects or the low effects in the population. So the disease phenotypes. Now we have the genes. Now we know the polymorphisms. Now we know entire information. Now we have the tools also. So because of this, now we are not going to use any of the position independent methods. So we are going to use only position dependent approach, because position we know that. So what is the gene? Chromosome 4 or chromosome 7 is having the ADH gene. This ADH gene we can just sequence. We can design the primers, we can just sequence, we can do the RFLP, we can identify the polymorphism, we can do anything. So that to discover the genes, both family and population based studies are designed. So identify the, for example, diabetes. If you want to study the diabetes, maybe you can just select some of the families who are having the both affected and unaffected family members, or maybe you can just take the population based studies for identifying the genes in the particular. So linkage and association methods are also used to analyze. So initially, if you want to identify, you have to identify the linkage studies or otherwise association studies. Linkage studies usually involve looking at the large families. For example, 5-6 generations family will be there. This family will be having the more affected individuals, and from several generations will be too. So that is called as the linkage studies. So linkage, success in the linkage studies is depending upon the Mendelian disorder gene mapping. So for example, sickle cell anemia, or the Mendelian disorder means cystic fibrosis, Huntington disease, or muscular dystrophy, where the gene is there, mutation is there, disease can be seen. That's called as a Mendelian disorder. So linkage identify the genetic marker that always inherited by the family members. So affected family members will be showing that particular gene, unaffected family members will not be having that particular gene. Or otherwise, more amount of, more frequently it is shown in the affected individuals, very less frequently shown in the unaffected individual. That's why we can just say about the linkage markers we use it to identify the disease. So linkage means, so you should go back to again your class 9th or 10th, where you can just see the cell division and where mitosis and meiosis is happening, and how the gametes are formed. Gametes are formed because of the meiosis. So one particular cell will be leading to the 4 different daughter cells. So in the daughter cells, maybe when compared to the parent cells, daughter cell will be having exactly half of the gene. And you can just see this in the meiotin. Actually, in the packeting stage, you can just see the exchange of genetic material. 2 chromatids, sister chromatids exchange the genetic material. So you can just see the recombination here. Recombination, that means shuffling of the genes will be occurring between the sister chromatids. Assortment of genetic loci, because you just remember the Mendel laws, assortment of genetic loci. So crossing over between the neighboring loci produce the recombinant gametes. So these recombinant gametes, how the recombination frequency is there in the mutate or the gametes, that only will decide what is the frequency of the genes that are present in the affected individuals or unaffected individuals. So recombination fraction we have to calculate. So for calculating the recombination fraction is, recombination means that is the measure of genetic distance. Genetic distance means one individual to the other individual. How much of genetic distance is there? One individual, how is different from the other individual? Yet what extent the genetic differences are there? Maybe it is a small single included polymorphisms or the deletions, whatever it is. So generally we just talk about the recombination fraction. We call it as the centimorgans, we say. Centimorgan means 1% of chance of recombination in the loci. So recombination between the two loci is 1%, that's called as 1 centimole. So minimum distance for the recombination is 1% of chance of recombination is 1 centimole. So generally we call this as a log score, log of odds ratio, log score we say. So log of odds ratio is to calculate the linkage distances. So linkage scores always reported association with the linkage frequency, that is the theta. So log score is equal to, that is called as a denoted with a Z, and log score is probability of the birth sequence given at linkage value and without linkage value. So with linkage value by without linkage value, that is the log value will be called as a log score. So recombination fraction, it is the measure for the genetic distances. So when the log score is more than 3, after doing the linkage study, the linkage study means we have to take the multiple markers from the chromosome and multiple affected individual from the family you have to recruit, and you have to sequence all the individuals for that particular markers. Sequencing or maybe microsatellite means genotyping you have to. So when you calculate the log score, if the log score is more than 3, you can just say that there is a evidence of linkage. So this particular marker is associated with the disease. If this particular gene is there, this particular mutation is there, the person will be having the disease. Perfect linkage, we can just say. If it is 2 less than 3 to 2, then maybe you can just say, okay, suggestive linkage. Maybe this particular region of the chromosome or this particular mutation may be linked with the disease. If it is less than 2 to -2, then uninformative linkage. There is no linkage. And exclusion, you can say less than -2, maybe it is exclusion. So this particular region is nowhere linked with this particular disease. So the level of or the value of the log score will be used for denoting the linkage or identifying the linkage after conducting the linkage studies. So strength of the evidence. Strength of the evidence, when you get a very good evidence for the linkage is if we have more families. And the families, we are having the more affected individuals. And you are analyzing the more markers, entire 24 sets of chromosomes if you are using, 23 pairs of chromosomes if you are using, even X chromosome and Y chromosome we separate it because the Y chromosome is exclusively present in the males and X chromosome present in the hemizygous condition. Only one copy will be there in the human being, and the males and 2 copies will be there. That's why I'm talking about all 24 types of chromosomes when we are analyzing. And we look for the more informative markers with a higher log score. Anywhere it will be there. It is not exclusively present in this one. So for the linkage analysis, you will be selecting the markers from all along the 24 types of chromosomes. So a series of linked markers will give the more confidence. So if more of linked markers are there, more confidence will be there, more. So whatever the, for example, 2.5 of linkage you have identified, that confidence will come with this particular number of markers, more number of markers. So generally what will be there in the marker means? Marker means it's a repetitive sequence. So CA repeats, CA will be there, CAG repeats, GATA repeats, GATA, because the repeat number will be varying. So for example, all our 35 members, if you take it, somebody will be having the 10 repeats, somebody will be having 11 repeats, somebody will be having the 12 repeats, and somebody will be having the 14 repeats. The repeat number will be because these repeats are abundantly present in the genome, and they also show a lot of variation in the repeat units. So 2 to 6 repeats, for example, CA repeats means 2 repeats, 2 nucleotide repeat, 3 nucleotide CAG repeats, or GATA repeats, pentanucleotide repeats, hexanucleotide repeats. So they are the repeat set sequences we'll be using, and linkage markers, for example, and we have used this particular panel for the cleft lip and palate analysis in 2007 to 2014. I have conducted a study for the linkage study in the cleft lip and palate using this particular markers. Markers will be present, exclusive Y chromosome we have excluded because it is not present in both male and female. So cleft lip and palate is present in both the things. That's why we have used the markers all along the 23 pairs of chromosomes, 22 pairs of autosomes and 1 X chromosome we have used. So you just see that each red color line indicates one particular position of the marker. So these many markers were selected for analyzing. Almost 360 markers were analyzed for the linkage panel. These linkage panels are available in the ready-made from the Applied Biosystem. You can purchase anytime. So determining the linkage interval, for example, first analysis, I have identified the linkage pattern here. And after that, what I will do is I'll just take some more markers from here, and I analyze, and I can extend this particular linkage extend. So I just expanded the linkage. Initial studies, I have identified only this particular region. After replication with the more number of markers from the neighboring regions, and my linkage region now established to little extent, more extent, and candidate genes. This particular region is having all the genes that are responsible for the cleft palate. Maybe if your study is for the diabetes, maybe you can just say that, okay, this particular region having the genes responsible for the diabetes, or maybe sickle cell anemia, or maybe some complication, or anything you can just say. So initial stages, you get maybe very narrow range. Maybe by identifying the markers or by analyzing the markers, flanking markers on the 3' side and 5' side, maybe you can just get more amount of established region for this one. And in this region, you can just explore for the candidate genes. Candidate genes means they are the set of genes that are involved in a pathway. Those genes are responsible for this. For example, alcoholism, you say that ADH is one candidate gene, ALDH one candidate gene. And like that candidate genes, that metabolism, ADH means alcohol dehydrogenase, ALDH means aldehyde dehydrogenase. So post linkage, you have identified one particular region on the chromosome number 7, and that particular region you have to identify, and that sequence you can just blast it by doing some bioinformatics analysis in the ANC PI browser or the UCSC genome browser. Maybe you can evaluate what are the genes that are present in this particular region and what are their functions. So what it will do, for example, ADH is identified. ADH, what it will do? ADH will convert the alcohol into acetaldehyde. ALDH will convert the acetaldehyde into the acetate. So these are the two genes that are responsible. Okay, chromosome number we have analyzed. So this particular gene is having this particular region. And start with the sequencing of the genes. Most relevant genes, you can just take it. And maybe if you don't have the money to analyze the entire region of the chromosome or the entire region of the chromosomal region where you have identified higher large score, maybe you can identify the genes, and that genes also maybe you can just go for the exons. Exons are only protein coding region. Introns are non-coding. So maybe coding regions you can concentrate, and then maybe if time permits, the money permits, or if you are having the sufficient funding, maybe you can just entire gene region maybe capturing for the sequencing. So finding a mutation and providing its causality is ultimate proof. So you have identified the alcohol dehydrogenase is involved in the linkage studies. So now we have to establish whether any ADH gene is there, that defective gene is there, the person is not getting the alcoholism. If the person is having this particular gene, he may be getting the alcoholism. So ultimately establishing the proof, cause of causality. So if this mutation is there, this particular disease will get. Any disease, not I'm talking about only just alcoholism. So any disease you take it, you identified one particular gene, for example, insulin receptor. Insulin receptor mutation is there, the person may be getting the diabetes. This mutation is not there, person is not getting the diabetes. So that's called as a causality, cause and relationship. So you have to provide. So for that, what you have to do now? So for linkage study, you have identified the region, and you have by bioinformatics analysis, you have established the region and identified the genes that are responsible or the polymorphisms that are responsible. Now allelic association. So this is actually linkage, extension of allelic association, the extension of the establishment. So you have identified now extension of linkage to the population level. That is actually a family. One family you have identified. Now you are going to establish the entire population level. So whatever the analysis you do, it should be having the wide applicability. It's not only for the only one particular family, my family or your family. It has to come to the population. You have to establish. So wherever the ADH is there, so that person, very fast ADH is there, that person will not become the alcoholic. That you want to establish. So what you do, you have to take the population. So population 1, when you analyze, so 6th allele is associated. 6th allele is frequently present in one population, and 5th allele is also present in another family. So then what you will say that, okay, 5 and 6 alleles are frequently present in the population. So population 1. And population 2, when you analyze, maybe you can find only the 6th allele. So there is no 5th allele. So in this population, you just see that some of the family members are having, or maybe some other population, some of the caste, maybe you can just, very caste specific populations also will be there. in the population. So here, when you analyze one particular region, maybe South India, when you analyze, you got that 6th allele and 5th allele is very popularly present or extensively present in the population. When I analyze the North Indian sample, maybe only 6th allele is present. So here 6th allele is associated. When you are publishing the paper, you will be talking about the 5th allele is also associated, 6th allele may be associated, like that. So these are the population differences. You can just see that this one, because these differences, what makes that is association in the 3 common forms. So direct association, when the mutation is there, when the allele is there, when the gene is there, maybe you can just say that disease is present. That is called as a direct association. Allele of interest itself is involved in the phenotype. ADH, inactive ADH is there, they will become the alcoholics. Active ADH is there, they will not become the alcohol. That is the direct association. So measure of the disease relevance directly by ignoring the other markers. So we are not bothered about other genes and other chromosomes, other markers. So we are just in taking the ADH we are taking, and ADH is directly linked with the alcoholism. So indirect association is there. Indirect association, we never correlate the allele with the disease. We always talk about the nearby marker that is causing the disease. The nearby marker, we take one more surrogate. So when the disease marker is present, one particular marker is present, that is called, for example, here M1, M2 is there. These are the regions, and this individual is having this N, then maybe you can just talk about disease present. So access to the trait is very difficult. That's why what we do is we take the other marker, correlated marker, that marker susceptibility we do, that is called as a surrogate. It is acting as a surrogate. It is not, that's called indirect association, we say that. So sometimes what happened is there is no association at all, but it shows as an association. So sometimes maybe get the spurious association. Apparent no association will be there. It is nowhere related with the etiology, but still you can get sometimes association because of the analysis or the population variations or something. Spurious. So association may be a spurious association, association may be indirect association, or maybe sometimes maybe it is a direct association also. And what are the different studies you need to design here? Maybe sometimes case control studies, sometimes family-based studies. So case control studies, when we are talking, case control studies are means we are not bothered about what is the population, what is the this one, what is the caste, what is the color, what is the sex, everything we will not see. What we see, whether the person is having the disease or not. So disease means case group, no disease means control group. Its technique is most powerful, this one. So disadvantage of this particular thing is population stratification can be seen. Population stratification means one particular population, as I told that 6th is present in one population and 5th is present and 6th also is present in other population. So that population stratification limit the case control conclusions, case control definitions and case control conclusions in that particular thing. So case control design is a much powerful, but it is having the problem with the stratification. Whereas the family-based studies, it is not, population stratification is not at all a matter here. Because this population, family-based this one, you will be having the disease and disease allele only in that particular family. So it is less powerful because very hard to collect the families, because it is very difficult to convince the multiple members, affected individuals in a multi-generation family. So family-based conducting the family-based studies is little difficult when compared to the, because if I go and collect diabetes, I can collect in 2 days, 50 diabetes, I can collect, and controls I can collect. But family-based, for example, convincing the individuals because of other social stigma and all other problems is very difficult. Family-based studies is not feasible in a long run. So allelic association you see here, and you can just see this when we are talking about the allele 6 is associated in the controls, it is very less is there, in the cases it is more is there, then we can just say that 6th allele is associated. You just see them very frequently present in the cases, 6th, very less frequently present in the control. Just we see that how many times it is present, very commonly present in the cases, then we can just say that we just associate with that particular thing. So the main blame in this particular thing is population stratification. Same thing if I want to do in one particular population, South Indian population, North Indian population, we take it. So South Indian population 6th is present, and North Indian population, that frequency is very less. So because of that particular population, even though it is associated in South India, it is not associated in the North India. So analysis of the mixed samples having different allele and different frequencies is the primary concern in the human genetics. This population stratification is very dangerous. So it leads sometimes false evidence for the association or the spurious association in this one. And the requirement is group differences in the allele frequencies and group differences in the outcome. Maybe some regions, one particular disease is more prevalent, some disease is very rare in the particular population. For example, sickle cell anemia, you take it. Sickle cell anemia is not present in the South Indian population. Very few areas of Andhra Pradesh, you can just see that one tribal population you can see. And if you go to the Nilgiri Hills in Tamil Nadu, maybe you can find Kurumba, Badaga, some tribes are there. And whereas you come to the Tropic of Cancer region, Madhya Pradesh, Maharashtra, Chhattisgarh, Gujarat, Odisha, these populations are having very quite common disease. More than sometimes 20% of the 20% of the carrier frequencies are identified in the populations. So that much this one is. So population frequency, here one small thing I'll just show you. Here what happens is, here two different samples I have taken. So first sample, the frequency is affected and unaffected, and this is the mutation present and mutation absent, wild type or mutation present. You can just see this, when you pull all the samples, you got the significance. So P less than 0.05 is the significance, you got the significance. Two different populations you have taken. So South Indian means very vague, you just take it because we have so many ethnic populations are there. So whereas the same study, if I conducted, you got their association. When you conduct the same thing, and here we got the non-significant, here we got the non-significant. We got, when we pooled all the samples, we got the significant. That was the population stratification. When one particular population only analyzed, no, it's not significant. Here one other population is not significant. When we pooled all the samples, then you got the spurious association here. So like that family-based association method, what happens? This will give somewhat better results because transmission disequilibrium test we perform. Who is contributing the disease allele? So our defective allele. For example, here father is contributing whether the maternal transmission or the paternal transmission, which transmission, who is contributing for this one. So 50% of the founders, either father or mother, will be having the disease allele, and they will be 50% of the chance for transmission and looking for the over transmission. We just see that who is transmitted. Who is transmitted more, whether father is transmitted more or the mother is transmitted more. These things only we just see. That is called as a transmission disequilibrium test we just conducted. And these studies are little rare in the populations people conduct, but they are much powerful than the simple association study like this. So here one particular population, for example, one community they analyze and they say that it's not associated. Other community they analyze and say that it is associated, or sometimes they may not associated. And when we analyze more samples without any population, this one restrictions, we get it association, that is a spurious association, because it is not the real association in this one. So advantages of this transmission linkage disequilibrium test is, or the family-based association test, may be robust for the stratification. Because here population stratification question is not there, because we are just looking only the parents and affected siblings, and robust for the stratification. Genotyping error is detected. So for example, sometimes after conducting the genotypes, maybe children, whatever the genotype is having, both the parents may not be having. Maybe sometimes they may not be revealing that they adopted child. Then after doing the genotyping, maybe you may not find that, because that is a Mendelian inconsistency, Mendelian error. So both father and mother is having different alleles, children is not having that particular allele. And sometimes paternity issues are there also, maybe you can find this stuff. So estimate the haplotypes is possible. So haplotype estimation is possible because you know that fathers found us. That's why you can just say that what are the probable alleles present in the gamete level. That's called as haplotype. Haplotype means particular pattern of polymorphisms at the gamete level. Gamete level, you can disclose it because of the father and mother, you know. children genotype, who has given which allele, that we can very clearly say in this one. That is called as a haplotype, estimation of haplotypes. So disadvantage is here, genotyping error you get. So after doing the genotypes, you may not get the proper this one Mendelian inconsistencies. You use heterozygous parents. We always get the heterozygous parents only we use it. Homozygous parents we will not use to identify this one. Inefficient for the genotyping for this one. And 3 individuals is needed. 3 individuals is needed means you need both mother and father and SIB, affected SIB you need, but 1/3 of the information only we will be using, either father or mother, who is contributing more, who is over transmitting, that only we just see that. Late onset disorders, psychiatric disorders, and pharmacological disorders is very difficult. For example, diabetes also, this population is very difficult because you have recruited the individual at 40 for study and that person is getting the 45 or 50 years diabetes. It's not useful. So late onset disorders, this is not at all useful, but this is useful for only for the early onset disorders. So this is one of the main disadvantages of this one. And the linkage, by this time you will be knowing the differences between the linkage and association. Linkage is the family based and association maybe is an unrelated population level also you can take it and family level also you can do it. Matching and ethnicity is generally unimportant. It's not required to match any individual. So age match, sex matched, matching need not be done here. And very few markers, 300 to 400 markers, microsatellite markers can be used in the linkage studies. Here, maybe sometimes 10 to the power of 6 SNPs. That means whole genome markers can be used. Its design is very powerful in the association, whereas the linkage design is very weak. And here, actually, initial detection for identifying the genomic region or the chromosomal region for the genetic association, maybe that is very much useful here in the linkage. Can identify. So this particular region, chromosome 13, 12.P12.5 is associated or linked with the gene. Maybe you can just take it. So powerful for the rare variants, you can just use it. Whereas here, family-based analysis, you can just take that unrelated matching. Matching should be done. And for example, you have taken from the cases from one particular population. For the same community, the control should be there. And you have taken 50% males, 50% females should be there. And if you have taken children, you have to take the children. You have taken adult, you have to take the adults. And these things should be taken. Powerful design will be there, but after initial detection for fine mapping, it will be very good. And powerful for the common variants and rare variants, it is not possible. Rare variant detection, you have to go for the linkage study. So designing a genetic study, because you should have a hypothesis. And hypothesis means what is your question? What is your for example, you want to identify the genes involved in the alcoholism, genes involved in the diabetes, genes involved in the PCOS, genes involved in the gum diseases or the bone diseases or some other diseases. What is your disease? Hypothesis. Research question is very important. And study design is very important. What you are going to design. So whether you are going to use the linkage panels and linkage based study, or you want to use the family-based study, you want to use the association study, population-based association study. So in that case, what are your samples? What are your genes? So these things are very much important. I hope that I have crossed my time limit. If you permit me, just 2 minutes, I'll just close it.
Sir, you can continue, sir, another 10 minutes.
Okay, sir. Then fine, I have very few number of slides remaining. So, okay, these are the very important. So, designing the study, because you need first hypothesis. Which disease you want to correlate with identify the genes for which disease you have to identify. Study design. So, these are all the, so how many number of genes you want to take, how many markers you want to take it. So, hypothesis means, here, first hypothesis means genetic study may refers to the hypothesis of the pathways. For example, oral microflora is causing the mouth gum diseases. So what are the different pathways? Host infectious agents related pathways, or what are the different genes, or bone morphogenesis genes, or some of the things, maybe hypothesized effects. So because of the oral flora, so there is a chance of getting the gum disease. So oral flora is again decided by the host genetic factors and hypothesized mediators. What are the different mediators? So how many times you use it to brush, and what are the different food habits, how many times you eat, whether after eating you will be washing your mouth or not. So these are the other moderators. They will be again deciding the factors. So genome-wide association study can also be used for the hypothesis generation. So if you don't know any gene earlier, maybe genome-wide studies also you can just take it, because more number of samples, you can just take more number of markers, you can take entire chromosomes, all 23 pairs of chromosomes, you can design the markers and you can analyze. So here just I have taken one hypothesis, that is the gum diseases. Maybe sometimes I might have given the lecture, this lecture for the genetics dental people. That's why I have one general, this is the general. How the oral microflora is contributing to the periodontal diseases. Periodontal disease means the individual's gum disease is decided by the several things. So oxidative stress, lipid peroxidation, other genetic markers present in the individual, tissue damage, and interleukins present in the individual, cytokine markers present in the individual, and all other things will be deciding the individual's inflammatory pathway. So hypothesized mediators are the individual's gum diseases are decided by the cortisol, stress levels, and biofilm formation, and COVID-19 and other infectious diseases. What is your gut microbiota and eating habits? What are the junk food habits? All the things will be causing to the death. And other moderators, you can just sit biofilm homeostasis and healthy oral tissues, and how your microbiota is going, what are the different healthy diets you take it. These things will be decided, because some of the moderator, not only the factors that are causing the disease is involved, and other moderates also will be responsible for the causing the disease. So some of the genome-wide association studies identified, these are the p-values that are plotted against the chromosomes. So each chromosome, higher p-values are identified in this particular thing. For example, chromosome number 1. So this particular marker is involved in the disease. So intronic SNP is identified. So this particular region, no need to do any linkage study here. All the markers from all the chromosomes were used for identifying this one. So wherever the higher p-value is there, you can just say that here higher p-value is there, here higher p-value. So wherever these markers are there, here also you can just say chromosome 11 also, there are some chromosome 11, chromosome 5, 6, 1. So these are the different markers that have been identified in the genome identity. So selecting the study designs here. So now the question is, the summary of my study will be giving is, as I told you that x-axis I have put the frequency, y-axis I put the effect, and I have told that the rare variant can cause smaller effect and higher effect also. Common variant also can cause very rare, very small effect and also higher effect also. So if the individual study, so very rare variant is causing the higher effects in the population or the disease, then maybe you can just design the family-based studies. So very common allele is causing the rare effects, then it can be identified using the case control studies. You can just take the diabetes cases and non-diabetes controls, you can just take it. So like that means rare, common mutation, common variation, common allele, very rare effects will be there. Very small effects it will be causing. That can be identified with this one. So very rare allele, very small fraction of contribution it is causing to the disease. Then you can identify it through the expression. Functional studies you have to do. So genomic studies, genomic functional microarray analysis can be done. So very common allele, very high effect it is showing. Then you have to use the cohort study. Cohort study, case control studies means, case control means cases and controls will be there. Cases will be having the disease and controls will be having this one. Cohort study means a group of individuals. So those who drink the coffee. So whether the coffee drinking is causing the peptic ulcer or not. So you have to take a group of people in a village. So individuals drinking coffee and individuals not drinking coffee, you have to recruit and make that effort, this one. And coffee drinking people, initially they don't have any peptic ulcers, and you have to watch them periodically after 6 months or 1 year, or 1.5 years, 2 years. So that means the repeated follow-up should be there. Case control studies is not like that. It's a cross-sectional study. So you can just recruit today Go there and diabetes identified, non-diabetics identify, you can just put them 2 groups and analyze. So cohort study is not like that. Cohort study is the group of individuals. So they are drinking the coffee, they don't have anything. And in the over a period of time, every time you visit, you identify some individuals developing the disease. So coffee drinking individuals will be having some people will be getting the peptic ulcer, and non-coffee drinking also some people may get the peptic ulcer. So who is getting the more? That we see. That's called cohorts. Cohort means a group of study, group of individuals sharing the similar characteristics. So those who born in 1974 is a group. Those who born in one particular village is a cohort. And a cohort means a group with a similar characteristics. So cohort studies can be used for identifying the larger effects with the larger alleles. So that is because of this one. Cohort design, I told you that cohort design, this is the cohort design. Exposure will be there and non-exposure will be there. Outcome will not be there in the initial stages. Peptic, nobody will be having the peptic ulcer. Peptic ulcer, you have to follow up and identify the peptic ulcers in the individuals with the exposure. That means coffee drinking. Non-exposure means non-coffee drinking individual. Both the individuals you have to see and who is having more, and you have to see that one. That is called a cohort study design. So here maybe at the end of this one, if anybody want to design, maybe family-based studies, you can just use it. Twin studies, adoption studies, pedigree studies, you can use it. Case control studies, cohort studies, functional studies, tissues, you can just take it. Periodontal tissues, you take healthy gingiva, a particular portion, and unhealthy gingiva, periodontal diseases tissue, cancer tissue, non-cancerous tissue, surrounding tissue, non-cancerous tissue, cancerous tissue. You can just take it and see, identify the gene expression profiles, and cancerous tissue will be having one particular profile, non-cancerous tissue will be having one particular profile. So those who are having the periodontal disease will be having one particular gene signatures, and other will be having the non-genetic this one. So like that, you can identify the genes that are involved, are apparently expressed in the disease individuals, not expressed in the normal individuals, will be giving the indication for the involvement of that particular thing. That will be decided based on this particular thing. So whether the allele frequencies contribution to the disease, whether the small effect will be there, larger effect will be there, low allele frequency or high allele frequency, those things only will be helpful for deciding the things. I hope that I can stop here. If you have any.
Thank you, sir. The floor is open for the participants for any questions. Dear participants, please interact with the resource person, Professor LVKS Bhaskar Garu, for any queries on his talk. Any of the participants, please respond.
Hello.
Yes, Gurumurthy Garu, carry on, sir. You are audible.
Gurumurthy sir, adagandi. You're audible, sir. You can ask the question.
You have to unmute it.
Hello. Am I audible?
Yes, sir.
Yes. Okay. Thank you for organizing this national seminar, and also thankful for this lecture by Dr. LVKS Bhaskar. He delivered a lecture on genetic study designs very well, and I know him. He's very dedicated and sincere. Personally, I know him. So I don't have any question. So I want to appreciate his efforts and dedication and hard work. Thank you, Bhaskar, for delivering this lecture. Thank you. That's all. Thank you.
Thank you, sir, for your nice presentation, sir. Thank you, sir.
On behalf of all the organizing committee, I profusely thank Professor LVKS Bhaskar Garu for his elaborate and vivid talk on designing genetic studies. And he has touched upon various aspects, right from the definition of the disease to heritability, genetic environment, genetic diseases, different types and types of variations, lifespan of the RBC, position dependent approaches, and ultimately he has given in the last slides, he has given vividly presented how to design genetic studies, family studies, case studies and control studies, cohort studies and functional studies. I profusely thank from the bottom of my heart on behalf of all the organizers for your elaborate and vivid talk, sir. Thank you, sir. Thank you so much for your
Thank you.
nice, wonderful presentation. Thank you, sir.
Thanks for the opportunity.
Over to Dr. Shanmugam Garu.
Sir. Now the oral presentations will be starts, and the presenters are Dr. Gurmurthy sir, Dr. Priyanka madam, and Dr. Rubin. Okay, so Gurmurthy sir, are you ready? Gurmurthy sir.
Hello, Shanmugam.
Sir.
Tell me anything to.
Sir,
nothing to respond now.
sir, అదే మీరు ప్రెజెంట్ చేస్తున్నారు కదా, ప్రెజెంట్ చేయొచ్చు సార్ వర్క్ని.
ఇప్పుడా? అంటే ఆఫ్టర్నూన్ కదా, ఐఎమ్ నాట్ ఇప్పుడు నాట్ రెడీ.
ఓకే సార్.
ఆఫీస్ వర్క్ లో ఉన్నాము, మీటింగ్ లో ఉన్నాము.
ఓకే. రూబిన్
చేస్తాను.
Rubin Sahu.
Yes, sir. Good morning.
Do you want to? Are you ready?
Yes, sir.
Okay, you can go ahead. Okay. Rubin, are you there? Rubin Sahu.
Hello.
Okay, you can start your presentation.
Good afternoon, sir. Am I audible, sir?
Yes.
Okay. So, good afternoon, everyone. My name is Rubin Sahu, and I am from Guru Ghasidas Vishwavidyalaya. And I'm going to present my work in the role of DNA methyltransferase genes in cancer induction, a systematic review and meta-analysis of DNMT1 and DNMT3B gene polymorphisms. So beginning with it, let me introduce you to the basics of the cancer. The cancer is a disease which arises from the uncontrolled cell growth due to disrupted cellular regulations, and it can be attributed to various phenomena ranging from the epigenetic modifications, which includes DNA methylation, and one of the prominent DNA methylation factors is the DNMT1 gene, which plays a significant or the crucial role in the gene expression. The DNMT1 is existentially responsible for the DNA methylation in the pre-existing DNA. Meanwhile, the two other subvariants of the DNMT, which are 3A and 3B, are solely responsible for de novo methylation, or carrying out the DNA methylation from for the newly synthesized DNA. And any the methylation pattern in of oncogene, specifically the CPG islands and hypermethylations in the tumor suppressor genes, contributes to the carcinogenesis. The polymorphisms in the DNMT genes we have studied and included in this meta-analysis includes RS1699593, RS2228611, RS2228612, and for the DNMT3B subvariants, the RS1569686, RS2424913, respectively. And they are known to influence the cancer susceptibility and overall leading to the cancer onset or the tumorigenesis. However, there are several individual studies which were found to be inconsistent in deducing a possible relationship with the cancer. And
ఏమంటే మధ్యాహ్నము 2 టాక్స్ ఉన్నాయి సార్.
in
this
study, we aim to perform a meta-analysis to clarify the association between these polymorphisms and cancer risk. So the materials and methods that we have utilized so far for conducting this meta-analysis includes, first of all, we have considered the PRISMA guidelines for conducting our meta-analysis. The database which we have searched for the papers or the significant data acquisition includes the Google Scholar, Science Direct, PubMed, up to 18th May of 2025. The last study that we have included so far was of 2025, and the keywords that we have explored include the cancer, DNMT1, DNMT3B polymorphisms, and the combination of all of them. And we followed a very stringent inclusion and exclusion criteria, where the inclusion criteria were solely inclusive of the DNMT1 variants, which we have already listed, and DNMT3B subvariants, which we also have enlisted before. The available genotype data were assessed for the case and control studies, and the stringent exclusion criteria was followed by excluding the insufficient or irrelevant data. Also, some of the data that have been acquired from the foreign authors, which were not readable, or in the foreign languages, which is not familiar, is excluded. The extracted details includes the author, year, country, ethnicity, genotyping methods, cancer type, and the genotype distribution overall, to add up to the credibility of the meta-analysis. The discrepancies which we encountered were resolved with the help of a reviewer. The statistical analysis were conducted for meta-analysis were performed using the MetaGenio, which is a web-based tool to carry out the meta-analysis. The Hardy-Weinberg equilibrium and the chi-square test were conducted to analyze the overall credibility of the data, and the heterogeneity was assessed using the Cochrane's Q test and I-square value, where I-square value greater than 50% contributes to the higher heterogeneity. And accordingly, we have equipped the random effects and fixed effects model for the pooled odds ratio, along with 95% confidence intervals. The results were visualized with the help of the forest plot, and the subgroup analysis and sensitivity analysis were also performed to ensure the robustness of the studies that we have made. Now, moving on to the results. So the study statistics that we have in this meta-analysis were 57 articles that we have identified, along with the duplicates that were also enlisted in that. So we removed 293 articles and screened them. The exclusion were solely based on non-cancer related articles. Some of them were the review articles. the duplicate data, missing SNP, and no control group or the lack of the control group. And hence, at the end, we were presented with the 54 studies, out of which the DNMT1 polymorphisms, RS1699593 has 13 studies, RS2228611 had 14 studies, and RS2228612 had 7 studies. The DNMT1 studies were mainly composed of breast, cervical, gastric, leukemia, and the prostate cancers. Predominantly in the Asian population, there were also other populations observed, but the Asian population was quite significant in terms. The DNMT3B polymorphisms that we have encountered includes 24 studies of RS1569686 and 25 studies of RS2424913. And in comparison to DNMT1, the DNMT3B covered the broadest aspects of the cancers, inclusive of the liver, lung, and colorectal cancer, apart from the breast, cervical, gastric cancers, and also included a good or broad aspect of the ethnicity, inclusive of the Asian, Caucasian, and African populations. So, the meta-analysis of the DNMT and DNMT3B gene variants and the risk of the cancers we have analyzed here is shown. The meta-analysis evaluated the DNMT1 and DNMT3B with the overall risk of the cancer. The heterogeneity was significantly observed in the studies so far, and hence we used accordingly the fixed effect models and random effects model. As we can see here, the recessive model of the DNMT1 was used to study the RS2228612. Where we got the pooled OR ratio of 1.20, which presents the significant risk of cancer association. Also contributing to the p-value is 0.28, which is quite insignificant to conclude these results. But the other models analyzed so far had presented us with the data that the DNMT1 polymorphism RS2228612 was significantly associated with the increased cancer risk in the recessive model. The DNMT3B variants, meanwhile, RS1569686 in both the allelic and the dominance model had shown to reduce the overall cancer risk. Also accompanied by it is RS2424913, which reduced the risk of cancer in the recessive models. Overall, the DNMT1 and DNMT3B variants showed mixed effects on the cancer susceptibility, where DNMT1 contributes to the enhanced risk of the cancer development, specifically the gastric cancer, which we'll see in the later sections of our presentation. And the DNMT3B variants, subvariants, which were solely responsible for the protection, for providing the protection from the cancers. So, in order to add up the robustness of the data, we also conducted the subgroup analysis. The subgroup analysis explored the effects as per the ethnicity and the cancer type. The DNMT, since the DNMT1 studies were heavily encircled around the Asian populations, and thus it limited the ethnic comparisons. The few per cancer type is a limitation. There was no clear or universal pattern which we can find out in the DNMT1. Apart from that, we also got the standout findings where the DNMT1 RS1699593 was deduced to increase the overall susceptibility of gastric cancer risk, which was significant in both allelic and dominant model. As we can see here in the table, that for the gastric cancer, the DNMT1 RS1699593 in the recessive in the allelic model, the p-value which we have observed is 0.020, and the OR and 95% confidence interval were respectively 1.13 with a range of 1.02 to 1.26 for the confidence intervals. The 1.13 value of the OR value, or the odds ratio, depicts that it is significantly associated with the risk of cancer. Meanwhile, the p-value confirms its presence, 0.02, which is far below 0.05. In case of the DNMT3B subvariants showing the population-specific protection, here the RS1569686 and RS2424913 were known to be associated with the decreased cancer risk in the Asian population only. And no protective effect was seen in the Caucasian and the African. Here we can also analyze that in DNMT3 3B variants, the RS1569686, the overall susceptibility rate of the cancer was reduced. As we can see here, the pooled OR value of 0.79 presents us with the significant protection. And this is also supported by the p-value, which is quite significant, 0.023. The DNMT3B variants in general were associated with lowering the gastric cancer risk and also the lung cancer risk. Moving on to our next section, which is the sensitivity analysis and the publication bias. The sensitivity analysis was conducted by sequentially excluding individual studies at a time, so as to add up to the credibility of the results that we have found so far. Since a single study is not influencing the overall result, and the inference that we got over here is that all the DNMT1 and DNMT3B polymorphisms remains unchanged, and hence no single study exerted a significant influence on the pooled outcomes. The findings were quite consistent and thus stable and robust. The publication bias was also analyzed using the Beck's funnel plot and the Eggers regression test. The funnel plot shows the symmetrical distribution of the data, and hence any publication bias could not be found here. No evidences of the systematic publication bias was observed, and the overall results are thus produced to be reliable and methodologically sound. So, to conclude the overall studies, this meta-analysis that we have conducted for DNMT1 and DNMT3B subvariants, the following key findings were reported by us in this paper. That DNMT1 RS2228612 is linked to the significant increase in the risk of the gastric cancer, as we have already discussed, with the p-value of 0.02. In contrast, 1699593 and 222861 showed no overall associations, and hence was not significant in terms of cancer induction or overall tumorigenesis. Variants 569686, 424913, overall protective effect to the populations, especially when it comes to the Asians. Despite all the study variabilities, there was no evidence of the publication bias, and there was also the sensitivity analysis that we have conducted so far didn't presented us with a significant study or the reading that we have analyzed is affecting the overall outcome. The cancer development is thus attributed by the epigenetic dysregulations, which is a significant functions of the environment over our genes, and the hypermethylation and hypomethylations are known to affect the onset of the cancers by inducing or reducing the tumorigenesis. The functional relevance of the variants that we have already discussed infers hereby. RS1699593 is known to affect the DNMT1 structure or function. RS2228611 influences splicing and protein expression. RS2228612 reduces the overall DNA binding efficiency, and hence induces the risk of cancer, especially the gastric cancers, in the subjects that we have analyzed using the paper. The DNMT3B variants affects gene regulation at the promoter regions, and the altered DNMT activity contributes to the aberrant gene silencing, and thus overall risk or the protection to the cancer. The strength of our meta-analysis was that the rigorous study selection and comprehensive analysis was carried out, including the 54 articles that we have screened and finally carried out our meta-analysis. Also, there were some limitations which is limited to representation beyond the Asian population. The center of our analysis was kind of based up to these Asian populations due to the lack of the heterogeneous ethnicity that we can analyze, and also there are various studies or findings that we need to robustly say that other variants are providing the significant protection or assessing the risk of cancer. So we can conclude here that DNMT1 RS 2228612 is overall contributing to the risk enhancing variant. And the DNMT3B variant is the potential protective markers. So I would like to conclude my presentation here. Thank you.
So good work, Robin.
Thank you.
And anything from the participants? Okay, then over to Shanmugam. Thank you, Robin, for the nice presentation.
Thank you, ma'am.
Thank you. Over to Dr. Shanmugam. Shanmugam, sir.
Okay.
Sir,
Hello. Next presentation is by Dr. Priyanka, Government Degree College, Autonomous, Nellore. Priyanka, go ahead.
okay, sir.
You can start your presentation.
Sir, wait.
Okay, no problem, no problem. Hello? Sir. Hello? Sir. లేదు ఈ అమ్మాయి ప్రియాంక తెస్తాం.
సార్ అది నాకు ప్రెసెంట్ అవ్వటం లేదు సార్, ఎర్రర్ అని వస్తుంది. దీంట్లో జాయిన్ అవ్వండి
ప్రియాంక
సార్. నాకు some it is not showing, sir. I will send my document. If you show, I will present, sir.
Okay, you can present, no problem, directly.
నేను మీకు సెండ్ చేస్తా సార్.
You can present directly, okay, no problem.
But it's not it's showing error, sir.
PPT, okay. Send me. I will try.
Error.
మనం స్టార్ట్ చేయొచ్చమ్మా, వస్తుంది.
సార్ నాకు రావటం లేదు సార్.
So good morning, Shanmugu. This is Sudhakar.
Sir, wait, sir.
Okay.
Sir, Priyanka, నువ్వు ఒక 5 minutes ట్రై తర్వాత చేస్తావు.
ఓకే సార్, ఆఫ్టర్ దట్ ఐ విల్.
Okay. Now, next presentation is by Dr. Sudhakar, SV Government Degree College, Govindasam Arts College, Tirupati. So, Sudhakar, sir, you can present, sir. Sir.
1 minute. Yeah, my document is. Are you audible? My voice?
Yes. You're audible.
Actually, no, my document is not uploading. The same issue. I am using the mobile.
Okay.
Yeah, can I explain directly?
Okay, no problem. You can.
Yeah, presently we are working on the Teulocarpus santalinalis hardwood extract. Yeah, myself, we are working on antioxidant properties and antifungal activities, sir. So here we are preparing the nanoparticles, silver nanoparticles. So silver nanoparticles, we already, these Teulocarpus santalinalis plant is all over, especially distributed in South India, especially in Tirumala Hills. It is very famous. So it is having many phytochemical values. The compounds, earlier study shows this limited work done on this Teulocarpus santalinalis. So in the present work, we are concentrating on the antioxidant properties. At the same time, we are concentrating on the antifungal and antimicrobial activities, sir.
సార్ మొబైల్ లోనే చేద్దాం అనుకుని చేస్తే అదే ఎర్రర్ అని వస్తుంది.
Okay, can I continue, sir?
Sudhakar, sir, you can.
Yeah. So in my studies, we found that this plant methanolic extract showing that MIC studies we done. So we studied DPPH assays, CBTS assays. So these studies found that it is having good antioxidant activities, sir, by using silver nanoparticles, sir. So at the same time, we got very good results in antimicrobial activity also, sir. I am sorry to show the results, sir, because it is not connected, sir. Yeah, sorry for that, sir. Hello, sir. Finally, we concluded that we are working on both anti-diabetic activities and antioxidant, antifungal activities, sir. So right now we finished this. We examined, we analyzed these silver nanoparticles of Teulocarpus santalinalis showing antimicrobial and antioxidant activities by quenching the free radicals, sir. So I'm sorry, sir. With that, I'm concluding, sir. Thank you.
Over to Dr. Shanmugam. Sudhakar, sir, this is a good piece of work.
Thank you, Dr. Sudhakar, sir, for your valuable contribution by presenting your research work. Now, I welcome Dr. Priyanka from Government Degree College, Women, Nellore, DKW College. Priyanka, you can present. Priyanka, are you there?
హలో.
Okay, no present చేయొచ్చమ్మా.
నా వాయిస్ వినిపిస్తుంది కదా సార్?
You're audible. You can present, Amma. హలో ఏమైంది
వినిపించడం లేదా సార్?
వినిపించలేదు అసలు.
అరే సార్ మళ్ళీ పెట్టారు చూడండి సార్. హలో.
Okay, now you are audible.
Sir. So as the age progresses, our daily fluid replacement should be 500 ml plus daily excreted amount of urine. So whenever this abnormalities is observed with respect to the replacement of daily fluid excreted with the fluid intake, so that should be that is a measure which we can find during the old ages when renal aging take place. So it is affected by mineral fluctuation and hormonal signals. As the age progresses, there will be decreased absorption. However, there are certain essential mineral ions that should be taken into consideration. Generally, in urine, sodium, potassium, phosphorus, about 20 to 30 mg is usually excreted in the urine. If it crosses that amount, so I have given actually in my presentation. So what are the dietary components that are available for calcium? Means dairy products which support bone and metabolism. That dietary intake should be 1 gram per day. So so these are to be certain considerations during the aging people. So magnesium, it is present in legumes and nuts. So it plays an important role in improving the insulin sensitivity. So insulin, we have to maintain our blood glucose level. In order to maintain that level, the magnesium ion plays a major role, and it is about 350 mg per day. Intake should be in case of aged individuals. And potassium, it is available in fruits and vegetables. So the intake should be 1 millimolar per kg per day. So it reduces the blood acidity in the blood. So during this, as the age progresses, this acidic nature of the blood is shown to be increased. And sodium intake should be 500 to 1000 mg per day. If the load, that is the renal handling. So we have to, as the age progresses, we have to understand the renal handling capacity and make our dietary changes as per that. In order to maintain the renal or kidney without stress, we have to take 500 to 1000 mg per day. So that is about the sodium, and it is present in processed foods and our salt. So next phosphorus. So it supports bone and teeth metabolism, and our dietary intake should be 8 to 12 mg per kg per day. So it is present in pulses and nuts. So this is how this in order to relieve our body from renal stress, we have to maintain our intakes as well. So these are the essential macro elements that are that should be monitored in case of aging individuals. So sometimes this non-essential elements like cadmium, lead, mercury, arsenic exposure. As the age increases, these non-essential heavy metals are going to accumulate in our body. So, due to the industrial pollution, contaminated water, food chain, groundwater pollution. So this is going to affect our body. Like it is going to cause severe chronic kidney disease, osteoporosis, hypertension, anemia, neurotoxicity, and renal fibrosis. So these are the considerations we have to take as per as there is a heavy exposure of this non-essential minerals, our body is going to be affected. So we have to take an alternative strategy in order to maintain our antioxidant levels in our body. So, how we can manage this kidney without having stress? So there are certain strategies, alkaline water management. So it can increase the urinary pH. Sometimes urine infection, everything occurs due to the acidity in the urine, genitourinary tract. So that should be that can be reduced by consumption of this alkaline water. So so that it can increase the urinary pH and improve hydration to the body. Next one is fungal based mushroom diet, mineral homeostasis. So edible fungal supplements are rich in potassium, magnesium, selenium, and bio active compounds. This antioxidant supplements enriched compounds like fungal mushroom based products can be taken in order to promote the gut biodata and increase the mineral homeostasis in our body. So these are the certain strategies. So in order to prevent our body from renal stress, especially with respect to the aging age, with the increase in age, and in order to prevent the structural, functional, and hormonal alterations. So these are certain strategies, and avoid more environmental toxic environmental exposure, and integrated with the nutritional supplements. This can increase. So there is, as the age increases, there is certain urinary inconsistency also. Sometimes they are prone to go some like that. So in order to prevent, we have to put certain strategies in order to protect our age old people. So with this, I conclude my lecture.
Thank you, Dr. Priyanka, for your
Thank you, sir.
valuable contribution regarding the renal stress. That is about the health. It is very
Yes,
good,
sir.
very good presentation. Thank you.
Yes, thank you, sir.
Any questions from the audience? Any questions? If no questions, we will go for the next presentation. That is by the Dr. Gurumurthy sir, Government Degree College, Vidavallur, lecturer in zoology, Wild Pod. Gurumurthy sir, are you there?
Yes.
Sir, you can present your work, sir. Gurumurthy sir. Gurumurthy sir.
Okay, am I audible?
Yes, sir.
Okay, okay. 1 minute, please. I'm going to share my screen.
Okay, okay, no problem.
Okay. Is my slide is visible?
No, sir.
Okay, thank you. Thank you. So, good afternoon to all. I would like to present my topic on utilizing organ on a chip system to predict multi-organ toxicity in clinical trials. It is a mechanistic approach to drug discovery. This workforce, that is the rate of drug candidates in clinical trials, is largely attributed to unanticipated human toxicity and lack of efficacy. And often it is undetected and by the conventional animal models. Instead of that models, we can use other modern technology, that is organ on a chip technology. It is emerged as a disruptive frontier technology in toxicology. So this offers micro physiological systems that can mimic or similar to the human organ architecture and fluid dynamics. This study explores the integration of multi-organ chips to predict cross-organ interactions and systemic toxicity. So this transition from single organ platforms to interconnected systems, that is from liver to heart, heart to kidney, and their potential to replace the supplement of animal testing in clinical trial design. The study concludes, my study that concludes on organ on a chip systems that provides a high fidelity and metabolic data that identifies and the reactive metabolites and their secondary toxic effects. So this study that explores the pharmaceutical drug discovery is currently facing an e-room law crisis. So it is a pharmacology terminology. E-room law crisis means the discovery of drug is gradual. It is similarly, it is lower when compared to the cost of the drug. That means the drug discovery is lower, but the cost of that drug is increased. So generally, that the drug cost is doubled in every 9 years since the 1950s. The new drugs increases exponentially despite technological advances. A primary bottleneck is the failure of animal models to accurately to recapitulate human physiological responses. Approximately 90% of drugs that pass preclinical animal testing fail in human clinical responses trials, frequently due to hepatotoxicity and cardiotoxicity that was not observed in vivo. So this technology is very essential. That is, organ on a chip technology represents the paradigm shift by combining fluidics with tissue engineering. So organ on a chip platforms create a biomimetic environment where human cells experience sheer stress, nutrient gradients, and mechanical strain similar to the human body. As the field moves towards body on a chip models, that focuses has shifted to multi-organ toxicity, understanding how metabolite produced in the liver might induce arrhythmia in cardiac tissue or nephrotoxicity in kidneys. 1 minute. Then in this technology, the predictive toxicology mechanisms are emerged. The primary advantage of this technology is ability to study the first pass metabolism. In traditional assays, the drug is applied directly to the target cell. But in this technology, the drug first interacts with the liver component, and where cytochrome P450 enzymes metabolize the parent compound, and resulting metabolites flow to the downstream organisms, so organs. So this is the methodology. So now I would like to conclude my presentation. So this experimental study simulations that is known for hepatotoxins that cause secondary cardiotoxicity demonstrate the predictive power of these systems. In single cell assays, transferrin often appears safe. However, in a liver and heart chip, the hepatic metabolism of the drug reveals the production of metabolites that prolong interval in cardiomyocytes, accurately mirroring the human clinical observations. Furthermore, these systems allow for the identification of biomarkers of early toxicity. By analyzing the effluent from the microchannels, researchers can detect a troponin T and concentrations lower than those detectable in traditional serum samples, providing the lead time for safety interventions during clinical development. So this technology that faces some future prospects or challenges. So despite the potential, several hurdles remain: the scaling loss and material absorption and regulatory acceptance. So scaling loss, ensuring the relative size and fluid volume of organ compartments accurately reflects human physiological ratios. And material absorption, the most common material can be absorbed small hydrophobic molecules, potentially shaking dosage data. And regulatory acceptance, formalizing the validation of organ on a chip data for FDA or EMA submissions. So this study that explores the multi-organ on a chip systems represent the frontier of biological toxicity. By bridging the gap between preclinical data and clinical outcomes, these platforms reduce the reliance on animal models and provide high resolution window into the human systemic responses. The integration of these chips into early phase clinical trial design will lead to safer drug candidates and more efficient pipeline for drug discovery. Thank you. Thank you for giving this opportunity. Thank you very much.
Thank you, Dr. Gurumurthy sir, for your valuable presentation. And also, new topic. This is a new topic, I think. Tissue engineering.
Yes, sir.
Nowadays,
Yes, sir.
it is very well focusing on the new drug discovery regarding the tissue engineering. Okay, sir. Very good presentation, sir.
Thank you. Thanks, sir.
Anybody,
Thank you very
any
much.
questions? Anybody questions? If no questions, we will close the session now. At 2:15, we will gather and start the next presentation. That is by the Dr. Shivkumar Aizer Pune, and one more presentation is by Dr. Sainath, Vikram Sunder University, Nellore. Okay, so thank you, sir. Thank you.
Okay, thank you.
Hello. Hello.
Sir, good afternoon, sir. I'm Chakrapani, sir.
Hi, good afternoon, Ali.
Sir, the session starts at 2:15, sir.
Okay, should I join at 2 o'clock?
By 2:15, okay, sir.
All right, okay, thank you.
Okay, sir. Thank you, sir.